Good morning and welcome to the morning session of the symposium Advances in Synthetic Biology. This symposium is being hosted by the University of Hong Kong team and co-hosted by HKUST and CityU teams. Along that, we also have three high school participants joining us today: the St. Stephen's College, Ho Chi Minh U Memorial College, and the project collaboration between Carmel Park U Secondary School, Moon Park Moon School, and Mrs. Wu York Wu Memorial College. I'm Maitri.
I'm Betty. We're members of the HKU 2020 iGEM team, and we will be your MCs for the symposium today. In our confirmation email, as well as the chat room, you can find the brochure of our symposium designed by the HKU CityU team. And parts of our symposium will also be streamed live online with the technical support from the HKUST team, and we will send you the YouTube link very soon in the chat room.
Synthetic biology, as a lot of you guys might know, aims to put theories into practice by merging scientific and engineering concepts to design and fabricate biological components and systems that normally do not exist in the natural world. With these products, synthetic biologists hope to push the boundaries of science and aim for the betterment of civilization. Using principles and methods from disciplines such as biotech, evolutionary biology, genetic engineering, and computer science, synthetic biology enables us to redesign organisms to tackle problems in medicine, agriculture, and manufacturing.
For example, in the field of medicine, advances in synthetic biology can lead to more efficient and cheaper drugs, with targeted therapies that attack superbugs, and they can also provide the cure to diseases such as cancer. In agriculture, genetically engineered crops can provide a solution to food shortages and vitamin deficiencies, whereas redesigned microorganisms can help us in the fight against pollution and help clean the environment.
The International Genetic Engineered Machine Competition, or iGEM for short, provides a platform to students from all over the world to help push the boundaries of synthetic biology by sharing their ideas in the form of innovative projects that build on its concepts to tackle real world. This year, we have 3 high school and 3 undergraduate teams from Hong Kong participating in the 2020 iGEM Jamboree. All the 6 teams will be presenting their projects during the symposium.
To get to know and understand more about the advances taking place in the field of synthetic biology, we will have 5 renowned professors and professionals from the field to give keynote lectures regarding their ongoing projects and experience.
During the morning session, we'll first be having 2 keynote speeches and then presentations from the Hong Kong high school teams participating in the 2020 Jamboree. We'll host a discussion session between the participants and the Hong Kong iGEM teams in the afternoon session at the end of the symposium.
During the keynote speeches and presentations, please kindly switch off your video and mute your audio. Once each presentation is completed, we will have a Q&A section during which you can raise your hand and ask your questions through the chat room.
Our first keynote speaker is Professor Heike Sedorov, who will be presenting on synthetic biological approaches for a more sustainable agriculture. Professor Sedorov is a professor in plant and microbial biology at North Carolina State University. She completed her master's and obtained her PhD degree in biochemistry from the University of Göttingen in Germany. Her research focus includes metabolic engineering to improve agricultural sustainability and increase oilseed crop yield. Automation, better varieties, improved management, and waste reduction to precision agriculture have increased yields in many countries. While genetically engineered crops have contributed substantially to these improvements, synthetic biology has not reached its potential impact in sustainable food and feed production. Professor Sedorov's team is currently working on new technologies to modify the plastic genome and regenerate homoplasmic crops. This technology will enable the generation of crops with better pest resistance and provide a platform for the fast and safe production of biopharmaceuticals. Professor Sedorov will present examples of new research paths that will further support more sustainable, climate-resilient food production systems using synthetic biology. Due to personal reasons, this session won't be streamed in YouTube.
time constraint, we can only allow 30 minutes for each team. We'll ring the bell once when time comes to 20 minutes, and twice when the time is up. Thank you.
So just an additional comment. A lot of you guys asked about the streaming. So we'll be streaming only certain presentations from the keynote speeches due to personal reasons. So amongst the keynote speeches, for the guys asked about the streaming. So we'll be streaming only certain presentations from the keynote speeches due to personal reasons. So amongst the keynote speeches, for the guys asked about the streaming. So we'll be streaming only certain presentations from the keynote speeches due to personal reasons. So amongst the keynote speeches, for the asked about the streaming. So we'll be streaming only certain presentations from the keynote speeches due to personal reasons. So amongst the keynote speeches, for the asked about the streaming. So we'll be streaming only certain presentations from the keynote speeches due to personal reasons. So amongst the keynote speeches, for the asked about the streaming. So we'll be streaming only certain presentations.
Okay.
Yeah, sorry about that. So for the afternoon session, we'll have Professor Wu and Mr. Phillips, whose speeches will be streamed to YouTube. And in addition, the presentations from the iGEM participants, both the high schools and also the undergraduate teams, will be streaming it live through YouTube. Okay. So today, the first presentation will be from the CityU WFN WYY team, who are working on the project Cutinases, the Terminators of Microplastics. Their project proposes to design an efficient, cost-effective way to degrade microplastics by incorporating the cutinase gene into a bacteria symbiotic to local bivalves, which can break down the PET in microplastic via filter feeding, and thus break down bioplastic to minimize damage to the environment. On behalf of the CityU WFN WYY team, Ricky, Fiona, Pius, and Hugo will now be presenting their project.
Good morning. We are Team HK CPU WFN WYY, and would like to give a brief introduction on cutinase, the terminators of microplastic. Before we start, I would like to introduce our team. As you can tell from our team name, we are a joint school team. Ricky, can you, next slide, yes. As you can tell from our team name, we are a joint school team from Camel Paku Secondary School, Tonghua Group of Hospital, Wonfordnam College, and Tonghua Group of Hospital, Mrs. Wu Yerkyu Memorial College. And this is our team photo. This introduction will include the project inspiration, symbol solution, our progress, modeling, human practice, and special acknowledgement. Microplastic and nanoplastic are plastic broken down by sun, currents, or wind. With at least 8 million tons of plastic end up in the sea every year, you can imagine how serious the problem of microplastic is. Thus, the problem of microplastic threatens both marine animals and us. The news of different marine animals swallowing or trapped by the plastic bag has entered the public eyesight more often. The reason is that marine animals misunderstand the plastic floating in the sea as their food, and hence the plastic will either consumed or trapping the marine animals, like the microplastic found in bivalves and plastic debris in bird stomach. Bivalves were well known for its superior water filtering capabilities. Thus, it works as an indicator to show how serious the plastic pollution is. About 4.3 to 57.2 pieces of microplastic have been found in mussels and oysters. These bivalves have shown a decrease in protein and lipid content. Moreover, as bivalves are primary consumer, the microplastic will propagate and accumulate in the food web. Eventually, the top predator in the ocean and human will be affected. Hence, we think of, is there any way to solve the plastic pollution through synthetic biology, like making the bivalves able to digest the plastic? Now let's move to the symbol solution.
Our project aims to use GM bacteria to produce an enzyme named cutinase to break down the microplastic, which are mainly PET, inside bivalves. In phase 1, we are going to introduce a plasmid into E. coli and test for efficiency of different cutinases. The design of our plasmid will be explained later. Next slide, please. Next slide. Yeah, thanks. We're going to compare the performances of three cutinases, LCC, TF-cut2, and HIC, and the effect of adding mediator such as 1-hexanol, which is said to be able to increase the stable time of the cutinase, will be compared. We will also investigate how the enzyme's expression site affects the efficiency of the cutinase.
As for the experimental design, all experiment will be carried out under the environment of pH value 8.2 and 30 degrees Celsius. The reason for such a setting is to simulate the ocean environment. As enzyme usually works within a narrow range of pH, such setting can help to find out which cutinase is the best when they work in ocean. A total of 8 setup will be carried out in 2 groups, namely HIC, LCC, TF-cut2, and control setup. The first group of setup will include their respective enzyme, PET film, simulated seawater, and mediator. The second group of setup will be the same, except no mediator will be added. This will help us to find out to what extent does the mediator help in stabilizing the enzyme. Also to find out which cutinase has the highest efficiency in degrading PET plastic in the ocean. A total of 3 measurements will be carried out. The first measurement is to measure the change of weight of PET film. This will directly show that which cutinase has the highest efficiency in degrading PET. Also, we will observe the PET film under the SEM for any structural changes. And finally, we will use the pH stat to record any change of pH value of the environment, as cutinase activity for PET hydrolysis was assayed using a pH stat to measure NaOH consumption versus time. This method allows a continuous measurement as the result can be directly seen from the pH stat.
In the second stage, we will introduce the GM bacteria that can produce the best cutinase as tested in phase 1 into bivalves and observe its effect on them. As we mentioned before, bivalves' growth is inhibited by the microplastic. Therefore, we will compare the fresh mass, dry mass, and the protein and lipid contents of the bivalves with and without GM bacteria inside. Besides, we may also compare the amount of microplastic inside bivalves' bodies at the end of the experiment. My teammates will give you an insight into our progress.
So let me present you our progress. First, we have picked 3 genes of interest, namely HIC, LCC, and TF-cut2. They have different base lengths, optimum pH, and optimum temperature, which may behave differently in a simulated seawater environment. We will pick the one that works the best in such an environment and progress with it. And here's our plasmid. First, we have the cutinase gene, and we also have a sumo tab to increase solubility because we are not sure if the cutinase is soluble in water or not. And we also added a signal peptide for enzyme secretion, so that the purification of cutinase is easier. Since the signal peptide we chose have shown to misfold enzymes in previous projects, a rigid linker is added to prevent misfolding. And finally, we added a his tag for protein purification. And here's our assembly. First, we'll synthesize the gene fragments. The yellow one is the cutinase gene, and the orange one is the backbone with sumo tag and his tag built in. And the blue one is signal peptide with the rigid linker. In the gene fragments, we have 3 restriction sites, which will be used in the next step. And the next step is digestion. We will use 3 restriction enzymes, namely SAL1, SAC1, and KPN1 to digest and produce the sticky ends. The sticky ends will then stick together in the next step, ligation, and we will use DNA ligase to stick the pieces together tightly to produce our plasmid. And my teammate will show you our progress in modeling.
Okay, let me introduce the modeling part of our project. We use the next slide, please. Okay, we use the software Mega to generate a phylogenetic tree of enzymes that degrade PET. As shown in the figure, cutinase from fungi is labeled in the blue bracket. Cutinases from mycobacteria is labeled in the red bracket, while the enzymes degrading PET from other bacteria, such as actinomycetes and proteobacteria, is labeled in the green bracket. Some cutinases from each cluster, which are circled, are chosen so as to guess the performances of other enzymes in the nearby branches. Cutinase from mycobacteria is not chosen because most of them can cause serious disease like tuberculosis. Side effect of the gene on the GM bacteria is not known. The next slide, please. Concerning the setting in generation the phylogenetic tree, we use a JTT model as the substitution model. This model corrects for multiple substitutions based on the model of amino acid substitution, described as substitution rate matrices. The distance between each sequences is calculated by the formula D equals to 2 times TQ, where T is the divergence time and Q is the substitution rate matrix. UPGMA is used as the statistical method. The distance between each sequences is found by merging the two closest clusters until only one cluster is left. Bootstrapping is also carried out to indicate the accuracy of the tree. 50 replications were done to find out the bootstrap value.
So now I'm going to introduce our human practice with you. First, we have released 2 questionnaires, and the first one is for the participants of the post-mini talk, which was held by the Hong Kong University of Science and Technology, City University of Hong Kong, and our team. And the first questionnaire is to ask their opinions of the talk. And then the second questionnaire opens to the public, and it is asked to the opinions of the plastic pollution and our project. And let me share some example of our questions. Question 1 and 2 mainly focus on the plastic pollution, and question 3 and 4 mainly focus on our project. So apart from the surveys, we are also going to meet with different advisors. Next slide, please. Yes, and in the local community, we are going to communicate with the Hong Kong Ocean Park and also the Agriculture, Fisheries and Conservation Department, called the AFCD. Since AFCD implements the regulation on the control of the plants and animals, and the Hong Kong Ocean Park have conducted in-depth investigations into marine environments, therefore, we believe they can give us precise opinions on our work and give more details about plastic pollution. Moreover, in the international community, we are also going to communicate with the Worldwide Fund for Nature and Greenpeace, since both of them have conducted in-depth investigation into the marine environment, including the marine litter. Therefore, we believe they can give us precise opinions on our project. For example, the damage we could do to the nature, and talk about the sustainability and also our ethical factors of our project, etc. And finally, it comes to the special acknowledgement part. The first institution we want to thank is the Revive and Restore. It sponsored our team for $5,500. And what's about the second institution? That is the SnapGene. It sponsored our team with 6 access to the SnapGene. And that's all of our presentation. And I'm going to send a link of our questionnaire in the chat room later, and hope you can help our team and fill our questionnaire. So do you have any questions? Please leave in the chat room and we'll answer it. Thank you.
Oh, thank you. Thank you for your presentation. Could our panel members please provide some feedback to the team? If anyone has any questions, please also post your question in the chat room.
So hi, can everyone see me right now?
Yeah, we can see you.
Yeah. Hi, so I'm Simon from Hong Kong U. I'm now a postdoctoral fellow in Professor Julian Tan's lab. So it's quite impressive for a joint high school team to manage to get this done in such a short time. My first question is how long have you guys been engaged in this project?
We have formed our team and discussed about our projects since March, if I haven't remembered wrongly. Yeah.
March. So 5 months. You managed to do this all experiment and contact all the other parties.
Yeah.
So regarding your project, I do have a few questions. So my first question is, maybe I've missed the first part. If you can tell me once again the function of cutinase.
It can degrade the PET into TPA and EG, and some types of cutinase can directly turn the PET into TPA and EG, while some will also generate the MHET and BHET.
So if I can simply understand that as a digestion of microplastic.
Yes.
And then in a very practical scenario, if you are going to apply it to digest the microplastic in the ocean, how does this method compare to the other current method? Like people always mention, try to recycle plastic. Yeah. So how's the cost of your method compared to those? Have you guys looked into that?
We haven't considered about the cost of our project as we haven't conduct deep surveys, deep research into it. So comparing to the current method to reduce the plastic, the current method most of them target to reduce the plastic consumption, and our project aims to reduce the microplastics which are already formed in the ocean.
Yeah, so I just imagine that there will be a common question when you guys are in a giant jam, because everyone, when you say this is a very useful method to solve a problem, people will ask about what's the cost of it and how does it compare to any other current method. Yeah. So if you ask me, I don't have the answer also, but yeah, you guys might have to look in the literature, look for some more information, and yeah, be able to answer that question in the giant jam.
Thank you for your suggestion.
And one more, because just regarding your experiment, I see that you said you guys did your experiment with the pH 8.2, is it?
Yes, pH 8.2.
Why is that?
Because I've done some research, especially from let me see. I remember it's from a certain organization that is set.
So that's a normal pH in the ocean, is it?
Yes, this is a normal pH in the ocean. It's around 8.2. It's from Australian Academy of Science. And then
that will be interesting to see, because if you guys are working with all the other association like Ocean Park, and you have something like that, it will be interesting to see if you guys can collect some sample and really test the pH to see if there's a real pH in the ocean. And also, I'm wondering if that's the optimum pH for that enzyme. Have you guys tried to optimize the condition or function of that enzyme?
I believe the optimum pH of the enzyme is not 8.2.
We do conduct the research about the optimum pH of our cutinase test. Ricky is showing the slide 20. Yeah. As shown in this table, the optimum pH is around 7 to 9. So we think it is okay to for the cutinase to work in such pH environment.
Another question that I can think of is, yeah, I understand that the pH in the ocean tends to be a bit alkaline. But when you are trying to digest the microplastic in the ocean, you are not going to just throw the enzyme in the ocean. So probably you will collect the microplastic from the ocean, filter it, and then you apply the enzyme to do the work. So perhaps that might not be the exact pH that you need to apply the enzyme. So one more experiment that I want to suggest to you guys is try to optimize the best condition for that enzyme, like temperature, pH, whatever.
Okay.
Yeah. Yeah, that's it for me.
Thanks.
Good job, by the way.
Okay, thank you, Simon. We also have other questions in the chat room. Can we have Hannah, also from our panel?
Oh yeah, hi, thanks for the wonderful presentation. So I have mainly two questions. The first one is about the human practice. So it's already a few months, 5 months into your project, and just now you have mentioned in human practice that you have collected surveys and maybe some interviews. I would like to ask what aspects of your project have you changed or modified after you collected these advice or professional suggestions?
And so first, thank you for your questions. And now we have already released 2 surveys to the public, and we have collected some opinions. Like some people said that it will very difficult for a project because it requires very high cost, and also we need to do many investigation on the BIFAF. And moreover, I said that we were going to communicate with 4 departments, but as it hasn't done yet, so maybe we will change our aspect after visiting the 4 departments. Thanks.
Okay, that's nice. The second question is, if I'm not understanding it wrong, you are going to put the engineered bacteria into the bivalves, or is it just outside?
Yeah. This is in phase 2. In phase 2, but in phase 1,
okay.
we are going to just test its efficiency first, and we'll gonna put them into bivalve, or as mentioned in before, or we may also collect the microplastic here.
Okay, nice. So you are temporarily using E. coli. Will you are you expecting to change the chassis or like use another bacteria to engineer?
We do we have some research on the bacteria that are commonly found in the bivalves. So in phase 2, we may also try to use other bacteria to produce this enzyme.
Yes, that's my point. So I see that the genes that you have found are not coming from E. coli. So it might be a problem that you try to express fungi genes or other higher organisms genes in a prokaryote E. coli. And maybe if you are going to express something in a prokaryote, you may like to express it directly in something that's already found in bivalves. Yeah.
I see. Why we choose E. coli is just because it is easy to handle in
I understand.
high school team.
Yeah,
so,
it's a first step.
Thank you for your suggestion. We may try to use other types of bacteria or other types of organisms to help us produce this enzyme to degrade the PET. Thank you.
Yeah, and in the real iGEM presentation, you can have already researched some of these model organisms or some commonly found by microbes in bivalves, so that it's just to complete the story and let the judging panel know that you have already thought of these problems. So yeah, better be well prepared for this question.
Thank you so much for your suggestion. Thank you.
Okay, so thank you. So next we have a question from Paul. So can the team repeat based on what criteria and do you choose your enzymes for your project?
Before we start the project, we try to find some enzyme that can degrade PET, and after our research, we found there's various enzymes such as peptase or lipase or in our project cutinase, and we found that one of the enzymes that's rarely used is the enzyme we are now investigating, the cutinase. But we are going, we are going to find the okay. As I have mentioned in the modeling part, we just try to find some cutinase or we just try to find some cutinase from each cluster, so we can predict the enzymes performances of nearby branches. Yeah. Can I answer your question? Sorry.
Yeah, okay. So let's carry on to the next question from Calvin. So Calvin said, like, he remembers that there are some previous iGEM teams who have done similar project about cutinase. So how can it be innovative when it comes to the competition of iGEM? Like, what's the points that stands out?
So I will take the question. The special point about our project is that in the future, we'll try to put some symbiotic bacterium, which is genetically modified, into bivalves, which are famous for its filtering capabilities. And this could be a long-term solution to microplastics pollution.
Okay, does that answer your question, Calvin? You can leave comments there. Okay, thank you. So next we have Kenneth. Maybe Kenneth, you can unmute. Yeah.
Hi. I just have some short questions. Basically, actually, Hannah and Simon asked most of the questions I wanted to ask. But I still want to ask the actual implementation. So have you been having previous research on whether cutinase's expression in E. coli and also how it degrades PET or microplastic in culture and also in other environmental settings, as you mentioned, right? It's very salty. It's like a salinity is used in ocean. So I'm aware that also your temperature used is 30 degrees Celsius, but that's not actually the physiological, like the actual temperature of the sea. So I want to ask, do you guys do any research on how the secreted enzyme could react in the salinity, and also how the ion would inhibit the enzyme? And is it an enzyme that is easily inhibited, or would you try to do any tests? And also, like Hannah said, E. coli is not very good in in ocean. It doesn't survive really well. And also, like previous research, is there like any in vivo expressions of cutinases already been like very well researched, or is it kind of a new field?
Maybe I answer your last question first, and I will pass and I think my teammate will answer your other question. For cutinase, and it is actually an old enzyme. I said as in recently, many people are focusing on investigating the pathways of degrading PET, which is pathways is very famous, but we try to investigate the activity of cutinase because in our research, some cutinase is able to digest the PET directly to its final products, e.g. and TPA, but the pathways can only need the product of pathways need to overcome another reaction to give its final product. So this is we think this is one advantage of cutinase over pathways. So although it is some research is also done by the past iGEM teams or other researchers, we would also like to do the research on this enzyme. Yeah, can I answer your question? Thank you.
Okay, so I think that is it for the part for this presentation. So let's move on to the second high school presentation. So thank you again for your presentation just now. So now we'll have the presentation from the HKSSC team. Their project is on production of PDRP CBD complex for recovery of palladium in e-waste polluted water. Palladium is widely used in industrial processes as catalytic converters and in biomedical applications as drug carriers. However, palladium is an environmental pollutant which is difficult to be recycled. In this project, SSC team modified 25 palladium-2 reducing peptide from a selection of known palladium binding peptides by fusing them to a cellulose binding domain 3 derived from Clostridium thermosalum, but with an aim to recover palladium from e-waste polluted water. Okay, so on behalf of SSC team, Curtis, Conrad, Nathana, and Karen will now present their project and discuss more detail regarding their project. So SSC team, please.
Hello, hi. Okay, so I'll start. So good morning, we are HKSSC, and today we will be presenting our project. Palladium is a platinum group metal. It is widely used in many daily applications, including electronics, catalytic converters, and dental care equipment. In 2019, 213 tons of palladium are produced worldwide. However, some palladium mines are starting to deplete, but the usage of palladium is increasing by the year, so the price of palladium is rapidly rising. With such a precious metal, it is not surprising that palladium recycling exists. In fact, in 2019, 97 tons of palladium is being recycled. That is almost half of all the palladium produced. Well, some of you may ask, that's only half of them, then where's the rest of them? Well, the rest of them could be found in landfills or in sewage sludge. These palladium could easily enter the environment and do damage to the ecosystem, as palladium is extremely toxic to aquatic lives and plants. There are currently many different palladium extraction methods. However, most of them are chemical methods, which involve using large volumes of harmful chemicals and organic solvents. We believe that the existing methods are not very effective, and we need a green and inexpensive alternative so that more palladium can be recycled. And thus, we have designed this project with the aim to design a palladium-specific binding peptide, which can reduce palladium-2 compounds into palladium nanoparticles. Nathan will now talk about our design in detail.
So in order to design a palladium-specific binding peptide with reducing capabilities, we have 4 library peptides selected after some literature review, and the first 3 was discovered through phage display. For the first peptide, it is one of the reported peptides which have an ability to reduce palladium without using any external reducing agents. Serine contributes to binding ability and tryptophan can reduce palladium. For the second peptide, serine and threonine are important for binding. This peptide mostly consists of many hydrophobic and hydroxyl-containing polar amino acids, which helps coordinating of PD2 compounds. And for the third peptide, serine and threonine are for binding, as I said before. Arginine residues can coordinate with palladium and have strong interaction energies. Histidine binds to palladium, and this was predicted by the Monte Carlo simulation with bond fluctuation model. For the last peptide, it is a A6C11 mutation of the last peptide. Alanine has a minimal binding and cysteine is a strong binder. The weak binding at residue 6 and strong binding at residue 11 can increase the efficiency. Instead of using single tryptophan structure like the one in the paper Chiu 2010, a double tryptophan design was suggested to be more effective. And also, an alkaline medium may be more effective as well. So these are the peptide sequences designed by our team with 7 amino acids. And these are the peptide sequences also designed by our team, but with 12 amino acids. To improve our structure, we have to study the coordination mode, identify the binding residues, and select peptides with optimal coordinating structure. And these can be achieved through molecular dynamics simulation. Because our peptide is short with only 7 and 12 amino acids, CVD is fused with peptide and undergo purification. Cellulose have advantages such as inexpensive, naturally abundant, and it is a chemically inert material. So it was used as the protein tag and stabilizer. Amino acids such as cysteine and threonine have a stronger coordination bonds with sulfur, and we expect the results with these amino acids anchoring more readily in the molecular dynamics simulation. We plan to use the palladium salts in the experiment after the analysis using program in designed experimental environments in literature review. Palladium ions forms square planar complexes, and PdCl4 2- represents 85% of palladium concentration, and therefore PdCl4 2- is used in molecular dynamics. The peptide is bound N-terminally with the CBD, and NHE1 and PST1 are the restriction enzymes, which is used to ligate with vector. All 25 constructs follow this structure, and the length ranges from around 450 base pair. Our vector used is PET blue 2 containing LAC-CA gene for blue light screening. And for the final product, CBD is ligated with the plasmid, and the length is around 4,000 base pair. Now, Curtis will talk about the methods.
So the strains we will be using in this project is E. coli NEB5 alpha and E. coli T7 express. This is the flow of the experiments we'll be conducting to clone the plasmid into the cloning strain. We will perform restriction enzyme digestion using NHG1 and PST1 to ligate our insert and our plasmid and our vector. We will then perform gel electrophoresis to check if our plasmid is of the correct size. If yes, we will transform it into the cloning strain using electroporation. We will then spread it on a plate with ampicillin and perform blue-white screening with IPTG induction. We will choose the white clones to perform colony PCR to further confirm if the cells contain our desired plasmid. These are the experiments we will conduct after the plasmid is cloned into the cloning strain. We will first transform the plasmid into our expression strain, which will then synthesize our peptides. After enough peptides are synthesized, we will lyse our cells using osmotic pressure, which involves creating a sudden and large water potential difference. After that, we will pour our cell lysis into a regenerated amorphous cellulose column, where the cellulose binding domain will adsorb to the cellulose. And after some washing, we can purify our peptide. We will also perform SDS page and Western blotting to check if the purified protein is our peptide. As there is a color change of yellow to brown when palladium-2 complexes are reduced to palladium nanoparticles, we could use the color change to analyze the efficiency of our peptides. For example, we could use a colorimeter to measure the color change in order to see the reducing efficiency of our peptides, or use a spectrophotometer and a calibration curve to determine our palladium nanoparticle concentration. As nanoparticles of smaller size are more efficient, we would also like to use an electron microscope to determine our size of nanoparticles synthesized. Corinne will now talk about our human practice.
Okay, so thank you, Curtis. So we have conducted a survey about e-waste, which has been answered by around 47 respondents. So from the first graph, the result has reflected the respondents' lack of knowledge about the importance of how to correctly deal with e-waste. More than 70% of our respondents just treat e-waste as normal waste. And in the second graph, we can see that nearly 60% of the people who were surveyed on e-waste recycling and do not know how to recycle e-waste. Nearly 40% have some understanding of it, and only 2.9% know it well. And 0.8% do not really care about that. Therefore, it is actually necessary to enhance the general public's awareness of e-waste recycling and increase their enthusiasm for centralized e-waste recycling. This is the primary task for the establishment of a complete system of e-waste recycling. So in the third graph, we can see that nearly 2/3 of the people agreed that the government should have more support on the e-waste recycling service currently. This is why our main objective in the human practice are to learn, to educate, and to acknowledge the public about how electronic waste can be toxic and have a large ecological impact. We have also interviewed stakeholders about their opinions on our project and how we may improve our design so it will be best for our stakeholders. This is one of our interviewees. During the pandemic, we couldn't actually have the chance to interview the environmental organization face-to-face, so many of them are through phone calls. Luckily, we have a chance to interview with electrical applicants component purchaser, Mr. Chan. Mr. Chan has given us some really good advice on how to really improve our project. So our stakeholders have introduced the methods of how e-waste is being disposed of, which are frequently used nowadays. And they are mainly going to be handling in the following ways that show in the PowerPoint. However, we can actually see that most of the methods are not very effective and also cause a huge damage to the environment. Thus, we have integrated these ideas and put them into our project. We aim to spread the correct way of disposing e-waste toward more people and show the ecological impact of illegal disposal. We have acknowledged our interviewees' opinion on our project. They have uniformly supported our project on the basis that it is cost-effective and that we can help the environment. Therefore, we have been extremely careful and intend to proceed with our research after the detailed consideration of bioethical reasons. So now I'm going to pass the time to Nathan to talk about more on the modeling.
So for the modeling part, I will first talk about the rate equations. We wanted to find out the impact of byproducts of tryptophan reduction on producing palladium nanoparticles. In the diagram, tryptophan binds with PD2 compounds, and tryptophan will donate electrons to a nearby metal ion, which is the PD2, and forms the tryptophyll radical. There are chances to form tryptophan, ditryptophan, and kynurenine, and tryptophan is the main product of reduction. The formation of tryptophyll radical is irreversible, and we still need experimental data to find out the rates in the equation to improve the modeling, and in order to optimize the experimental condition and maximize the production of palladium nanoparticles. And this is the graph of the rate equation, assuming every rate is constant using the MATLAB simulation. And we'll fit in the experimental data in the graph once we got them. So Conrad will now talk about the molecular dynamics simulation.
As mentioned before by Nathan in peptide design previously, we are going to use molecular dynamics to screen our peptides with optimal coordinating structure with anchoring sites near tryptophan to reduce the PD2 compounds. However, molecular dynamics packages may not have all the parameters defined for the atoms added or the ions added inside the molecular dynamics simulation package. Parameterization of the force field are required to perform the MD simulation. The base of a molecular dynamic system was peptide structure and topology, and our peptides are processed through the PEPFO3 peptide structure prediction server. Out of the 100 structure generated, the structure with the lowest SOPEP score was selected and proceed with the simulation. The molecular dynamics simulation package we used was GROMAX 2020.3, and the force field we used are CHARM27. And the peptide we used in the preliminary simulation was the first library peptide. As I said before, modifications to the system files are required for the molecular dynamics simulations. We have added the residue name information in the root directory of the GROMAX in the residue types.dat file, and the addition of the PD atom, including its mass information, atomic mass, to the CHARM27 root directory in the atoms type.atp file. Residue topology informations are required to read the PDB generated, the PDB of PDCl42- ion to transform into the GROMAX trajectory file using the function gmx to PDB to gmx. Also, Lennard-Jones parameters of the PD and Cl inside the PDCl42- ion are added inside the FF non-bonded.itp. As information about adding the atom types and residue types information are readily available across the internet, I will focus on how to generate the include topology or the residue topology file. So in the first line, the bond types directive defines the number of bonds for the PDCl42- ion. The second directive, PX, defines the residue name for the use of the PDB file generated by the open Babel and inside the topology file. And in the atoms directive, it defines the atom name and the electron charge of the PDCl42- ion. In the bonds directive, it defines the bond length in nanometer and the bond stretching harmonic harmonic force constant of PdCl4 2-. Lastly, in angles, it defines the bond angle in degree of the ion. All the values added inside the residue topology file and in the include topology file are from previous literature. One thing special about the include topology file is the molecule type directive, which defines the molecule name. And in our case, it was px, which is the ion. To summarize, the generation of molecular topology for the PdCl4 2- ions with the atom information, bonds interactions, bond angle, and the Lennard-Jones parameters. So after we have generated the molecular topology for the PdCl4 2- ions, we are headed to the solvation of a system with our peptide. The water model used in our simulation was SPC216, and the size of our simulation box was a cubic box with its width, length, and height all being 6.051 nanometer. And the whole system was solvated by 7,206 SPC water molecules. This was a picture of our solvated system with the PdCl4 2- ion and peptide. After solvation, our system was in the charge of -1. And before we proceed into the energy minimization and the equilibration of our system, the whole system must be neutralized. Therefore, the system was neutralized by 1 sodium ion, as shown in this picture. So we have prepared our neutralized system and the solvation of system. We can now proceed to energy minimization of our system. One of the modifications to the options file of energy minimization file was that the electrostatic interactions were calculated by the particle mesh Elwood PME method and with a cutoff range of 0.9 nanometer. This was the result of our energy minimization. From the graph, we can see that we have successfully minimized our system's energy. So after energy minimization, we can now proceed with temperature equilibration, which was the NVT example of a system. We have ran the temperature equilibration for 100 picoseconds, and this is the result. So after temperature equilibration, we can now proceed to the pressure equilibration, which was the NPT example of a system. All the settings were kept default, and the equilibration was ran 100 picosecond also. This was the result. So after energy minimization and the equilibration of our system, we can now run the molecular dynamics simulation. However, there are some parameters we need to modify in order to carry out more accurate results. For example, Link's algorithm was applied to constrain the length of the hydrogen-containing bonds, and the frequency to update the neighboring list was 10 per step. After all the modification and the preparation of a system, 1 nanosecond of the simulation were performed. This is a picture of our molecular dynamics option file. So here is our result. So from the simulation, we can see that the arginine amine group NH1 in the picture and the PD of our PdCl4 2- ion have a distance of 2.87 angstroms apart. And through literature, we know that arginine having the strongest interaction energy with palladium and have a preferred distance between them as 2.9 angstroms apart. Therefore, we think that we have achieved preliminary success of our molecular dynamics, as our arginine's amine group was the group with the shortest epsilon, which was the distance between them. However, our molecular dynamics simulation, we haven't finished our molecular dynamics simulations. As mentioned by Nathan in the and Curtis in the experimental design section, our experimental conditions will be in alkaline pH. And since the PEPFO3 structure prediction server assumes the peptide structures are assembled inside a neutral condition, we need to change our structure in order to get a more accurate result. Also, due to computational power, we lack of computational power, we cannot produce more results. And the simulation of the rest of the peptide are time consuming, and we need to finish all the simulation of the rest of our peptide in order to get a full analysis of all of our peptides. And I'll now pass the time to Curtis about a proposed implementation.
So our proposed end user of our project are the metal recycling industry, the automobile industry, and the electronics industry, because we believe that these industries are the easiest to come across old parts or other materials that may contain palladium. So we could have them use our project to help recover the palladium and reuse it. Now I'm going to talk about the actual design of our implementation. We have designed this with making this project a industrializable and large-scale system in mind, and we have designed this. So the first step is to have a bioreactor containing our E. coli with our desired plasmid, which can produce our peptides. So the second step is to pour our supernatant, which is the cell lysis we have harvested, into a regenerated amorphous cellulose column, which I have mentioned before is used to purify our peptides. Then we'll put them into a tank with the metal ion solution. The metal ion solution can come from those old parts, as palladium could exist in palladium-2 complexes in those parts, and this solution may often contain other metal ions. Our peptide can then bind to our palladium-2 plus ions and reduce them into nanoparticles. However, we are still unsure if our peptide will reduce other metals, as we haven't done any experiments this year. Finally, we will put all of them into a centrifuge to separate the palladium nanoparticles and other impurities. We will repeat this process a few times. We believe that this system is generally safe, as we do not find any obvious hazards, except from the exception of the leakage of palladium. The leakage of palladium is possible in this system and may pollute the environment. We are currently facing a few challenges in this system, and we are aiming to overcome it. The first one is the solubilization of palladium. As not all palladium found are in palladium-2 complexes, some may be in elemental palladium alloys, and thus we need to separate them and solubilize them. We are also concerned about the specificity of our peptide and the purity of our nanoparticle, as we couldn't do any experiments to check if our peptide will bind to metals other than palladium.
Okay, so firstly, thanks for the support for our advisor, Professor Chen, and of course, Dr. Ho. And also, thank you, Mr. Shen, for providing the sequence of cellulose domain. Moreover, we cannot forget our student advisor. They give us really great support. They are Wang Zhonghin, Zhong Junho Anson, and Li Hong Hu. Last but not least, thanks for the support from SnapGene, New England Biolabs, and MathWorks. So now this is the time for the Q&A section. Thank you.
Right, so thank you SSC team for your presentation. So as if we have already some discussion in the chat room, so maybe let's start by addressing Marco's question. Can Marco unmute yourself maybe?
Hi.
Yeah.
Hi.
Hi. Great work. And
thank you.
In fact, I like your team. I like your team's dynamic. That's great. I just checked the price of palladium, and I found it is even more expensive than gold.
Yes.
So in fact, there are quite good commercial aspects of this project. What is the key of this project to be commercially viable, in your opinion?
The key, I believe that the key is to have a system that can pump out enough peptides, and for the peptides to be efficient enough to actually make this a viable system.
Also, I think that our system need to be easily adapted by the industry, as we aim that if our system are easily accessible and adaptable to the industry, it can really help the industry to recycle the used parts and palladium for their reuse and the help of to reduce the price or the other commercial applications of palladium.
Yeah. Have you guys cloned the construct yet?
No,
Due to the-
fortunately, no.
Yeah.
Unfortunately, no. We could not have any access to our lab this year.
Oh, yeah.
Yeah.
And this will be in our phase 2 project. Yeah.
Okay,
And all
good
the-
luck for that.
Okay.
That's all for my question.
Thank you. Thank you. So now we have a question from Dr. Yun from HKXUI team. So do you want to unmute yourself? Or maybe I can ask the question. So how do you compare the efficiency and cost effectiveness of your project outcome to the existing methods, both biological and chemical, of clearance of PD?
At this point of time, we could only compare it through literature. We know that tryptophan could reduce palladium in 4 hours. However, there could be other preliminary work that may require more time. However, the chemical methods would require a day or so because they would need to separate palladium with other metals first. Yes. So as to the cost effectiveness, we could see that many chemicals used are quite expensive, which includes rare organic solvents and large amounts of acids. Yes.
Okay, then can we have Kenneth and Hannah to ask your question?
Hi, I just want to ask, actually it's mentioned in your challenges, right? So basically, like the first thing is you need to separate the palladium from the solid phase, from the electrodes, or like from the industrial waste. So how is it better than the chemical methods? I mean, you need to do a chemical method first, and then you have a metal mixture that is contained with like a lot of metal ion. And you also have a lot of problem with the selectivity and also the exhaustion of peptide if they also bind to other metal ions. And so how do you exactly like provide a better method for purifying palladium and also making nanoparticles? And yeah, also about your peptides, right? I want to ask the nanoparticle. So you just want to do a peptide around the nanoparticle, like a wrapping, and produce a nanoparticle. Is there like other functionality of it? And is there like conjugation to other metal ions to produce more more different function of nanoparticles? As I look through online, there are a lot of research actually on palladium nanoparticle productions with these kind of peptides. So how is your project better than like designed to be novel and better than their research?
One thing we would want to stress is that most of the palladium nanoparticle research are required the external reducing agents. And in our project, tryptophan, this amino acid have the special ability to reduce palladium itself. And this will elevate the problem of the additional external reducing agents used in the production of our nanoparticles. And
as to the first part of the question, like the chemical part of the solubilization, we're currently exploring materials that could be synthesized by bacteria that could potentially leach palladium. Yes, some candidates, if I remember, was thiocyanate. Yeah, but then this part is still in a research phase, and we need more information and research to support this.
Yeah, so basically, like found the 2012 12 paper, also using a peptide with tryptophan that is surrounding a palladium ion to form a nanoparticle. And also they use the peptide sequence as also as a ligand and also as a linker to other metal ions. So how is your project better than the other research team that do the same thing?
One thing we also did in our project is the molecular dynamics simulation. And from the molecular dynamics simulations, we can try to simulate the structure and how it performs in the molecular simulation. Inside the simulation, we can predict its anchoring site, or if we can now predict with and try with different structures and try to find the optimal coordinating structures and how to improve the structure of our design. So this will be one of our goal and our directions of our project. Hope this answers your question.
Okay, thank you. Hannah, do you have any questions?
Yeah, most of them are. I mean, you have already answered the question in the chat room, like in the implementation part. But I think overall the presentation is very well, and I appreciate that you have done so many dry lab stuff outside the lab so that you make good use of your time without doing any lab work. And yeah, but maybe for the presentation, you can consider put the implementation part first so that everyone has a big picture of what you are going to do, and then you'll introduce the small parts one by one. And yeah, I have only one question regarding the technical aspect of your project, and it's about if you are going to test in the lab about your binding efficiency of the design peptide and like the binding between your design peptide and the ions, what will be the experiment? Say if you can go to the lab now and do it.
So to determine, did you mention binding?
Yeah, it's binding.
Especially binding.
So for the binding efficiencies, we may not have a well-designed experiment.
So you have designed a bunch of different peptides.
Yes, but for wet lab, we don't think we have a well-designed experiment for that. But for molecular dynamic simulations in a dry lab or inside or the in silico simulation of our peptide, I think that if with our results like the amino acids having strong interaction energies or having better optimal coordinating structures with our peptide and the ion, I think that that can somewhat represents inside how our wet lab results may reflect. But we really don't have some wet lab experiments to prove this concept right now.
Well, there is a rough thing we have seen yesterday, which involves having an NMR spectrometry to measure the binding studies of our peptide. So this could work, but we would still need to do further research into this part. Yes.
Okay, I see. Thank you.
Thank you. I think we have one more question in the chat room from BL. What is the specificity of your method in removing PD to ion, any metal ion interferences?
So again, to this question, we could only refer to previous studies we have read. So we could see that those peptides have approximately 70% specificity to palladium and rest to the platinum. So as to like the very accurate results with other metals, we would still need to do our own experiments in order to know that. Yes.
And also add some.
Hello, we cannot really hear your question.
Can you hear me right now?
Yes.
Yes.
Okay, good. So yeah, it's good to see another project trying to save the environment. So actually, Hannah asked the same question that I want to ask. Is about the experiment, how you're going to test the binding of your designed peptide to palladium. But she asked that question. I'm not going to ask that again. But in your previous slide, you showed everyone that your peptide, the amino acid tryptophan, is supposed to reduce palladium, isn't it?
Yes.
So if the objective of your project is to recycle palladium, and how are you going to retrieve that from its reduced form?
So referring back to the implementation, I think that Curtis can explain more on this slide. Curtis.
If you are talking about how do we separate the nanoparticles with the peptides, we could try using centrifuge, and if it does not work, we would have to rely on other chemical methods. However, our main concern is not on the bioleaching of palladium, is actually to create the nanoparticles which have wide use from unused palladium parts.
Also, there are some, we have read some research, reports on using centrifuge to retrieve nanoparticles. So that I think that the centrifugal method is viable via, we can industrialize the centrifugal method in our implementation.
The centrifuge is just using physical force to separate things. But the palladium is in its reduced form. So is it the objective of a project is to apply that reduced palladium or for something else? But
yes, we are just providing a method for the maybe the automobile industry to retrieve back the used palladium and then recycle the palladium and use it again inside other like catalytic converters, or use in other electronics to make alloys with other uses. But because some like e-waste, maybe they are not, they still contains palladium, but some of them may not be able to be reused again. So therefore, a method for retrieving the palladium are needed for the industry to retrieve back and recycle the palladium uses.
So the ultimate goal is to get palladium, but not the reduced palladium.
Yes, basically.
So it goes back to my question. If you're just using centrifuge, you're not doing anything to the chemistry of that palladium.
This is after the reduction. Our main goal is to make palladium nanoparticles from palladium tetrachloropalladiate, and the used palladium should be performed with bioleaching, and that is not the main concern here, because after bioleaching, most of the recycled products are still in the iron form, and we wish to change the iron form back to its elemental form, and this is after the reduction. So it is a mixture with palladium nanoparticles and our peptides and some unreacted PDCl4. So we could use this to separate. And
the reduced form of our palladium is already in the PD0 nanoparticles. So we retrieve back the nanoparticles, which is the reduced form of the palladium in our experiment.
Well, it would be great if you can explain these in the very beginning, together with these picture in the implementation, because I guess quite a few of us are confused when you guys are presenting, because we are not sure how you're going to apply your theory. So it would be good to give everyone a whole picture with these pictures in the implementation. So you move these to the beginning of your presentation.
Okay.
Yeah, that's it for me. Thank you.
Thank you. I think we have another question from Professor Tim Chow. He asked, what is the selectivity of the peptide for the palladium and the capacity?
I think the question has already been asked, but then I think I want to add in is like about the capacity of how much you would be able to retrieve if the concentration is low, and then how much peptide you need to produce in order to be able to retrieve a specific amount which is useful.
So in our modeling part, we need to first study from experimental data. We can in the rate equations slide, we have explained how we can use experimental data to model a system and to see out the tryptophan's reduction capability or other things. That is like the test for the capability of our peptide, and therefore the use of the experimental data to optimize a better model for the use in the real world, such as in your situation to retrieve palladium in a low concentration. We need to optimize from the experimental data to a real world situation.
Mm-hmm. But I guess the point is that whether is economically viable in a way, and what would be the tipping point, whether it should be the approach to be taken.
We could see in some past research that low concentration of palladium can be reduced by a relatively low amount of peptides, which is about a few milligrams, if I remember correctly. For quite low, it's like.
However, the incubation time maybe not very well explained in this slide. Binding is not instant or a very quick action. It is maybe taking days or even hours or even days to complete the binding. So in low concentration, we really need to optimize and maybe study the economical or the cost effectiveness of our solution after we may have the experimental data to really know how our designed peptides having the capabilities to absorb the palladium ions. Or yes.
That is also a reason why we wish to perform the chemical kinetic studies, because we can, after then we could clearly understand the effects of concentration and how the concentrations impacts our efficiency and the whole system of our reduction process.
Thanks.
Okay, thank you, the SSC team, for your presentation. And does anyone else have any other questions? If not, then, oh, okay. Okay, I think Janet gave a comment in the chat room. Need to really justify why do you need to go through nanoparticle stage for purification instead of using very established chemical methods.
Sorry, we cannot really hear the question.
I could hear it. So I know it's a comment, but I'll just try to answer it. We're trying to use as little chemicals as possible. So that is our aim, to provide a greener method. So that is what we are trying to do. Okay, so thank you. I guess that's the end.
Okay. Okay, thank you, the SSC team. So we'll now have the presentation from the HCY team, who are working on the project Enzymatic Biofilm Degradation with Engineered Organism to eliminate biofilm-induced biocene resistance contaminations in biofuel and diesel tanks. Through this project, the HCY team aims to combat contamination in biofuel and diesel tanks. Treating contamination in these fuel tanks is difficult due to the formation of biofilm. The HCY team aims to degrade these biofilms by engineering certain organisms to release biofilm-degrading enzymes such as A-amylase, R-chymotrypsin, D-nasal, and certain protease, which can eliminate the biocene resistance contaminations in the tanks. On behalf of the HCY team, Kelvin, Cindy, Suki, Jackie, Angel, and Teresa will now be presenting their project.
Yes. Okay. Hello, everyone. Before we start, I would like the audience and the panel and the mentor to visit a website and to join our to join our session, which is the menti.com, and you can enter the code to get in the room, and then after that you can switch back to Zoom and listen to our presentation. So I would like all of you to do this step first, and yeah. Okay. Okay. I suppose all of you have get in the room in menti.com, so I would like to start my presentation now. So good afternoon, good morning, everyone. I'm Calvin, and we are from the Hong Kong HCY team. We are glad to be here, and our team is going to have further research on biofilm degradation and biofuel. And here is our content page. We are going to begin with a definition of biofuel and biofilm. Then we will talk about the current methods of biofilm degradation, and it will be followed up by our target enzymes and microorganisms. There are 5 steps in total from A to E, and it will help you familiarize with our project. Let's follow our path and get to know more. First, our proposed idea. Our proposed idea is biofilm degradation in specific enzymes released from certain engineered microorganisms. So let's start from introducing our first key idea, biofuel. So what is biofuel? Biofuel is a fuel that is produced through contemporary processes from biomass, rather than a fuel produced by the very slow geological processes involved in the formation of fossil fuels like oil. Let's give a local example of a biofuel production company. The Chamway Company in Hong Kong produces biodiesel from used cooking oil, UCO, and they have been working with a lot of big brands like McDonald's, KFC, Cafe de Caro Group for more than 5 years now. Also, more than half of the 4 to 5 stars hotel in Hong Kong provide UCO to them as well. Strangely enough, collecting UCO in Hong Kong is considered to be a service in which the company has to compete with other collectors.
Sorry, Calvin.
Yes.
Our PowerPoint is not moving.
Oh, because I'm not moving. Yes, it's not moving. Okay, we can continue the presentation. Can you see clearly now?
No, do you want to try making it to a presentation mode first? Like make it full screen?
Oh, I make it full screen, but maybe I
Yeah, maybe
tried.
stop share and share again.
Okay, I'll share again.
Thank you.
Okay.
Can you make it full screen now?
Can you see the full screen?
No. No.
Oh, why?
We can make another team member as co-host, so he or she.
Can Kathleen to have to present the screen? How about if I switch like this, can you guys?
Yeah, we can see.
Okay, then I maybe don't make a full screen, but use it by hand. And let's continue our presentation of Chinmay Company.
Thank you.
Yeah, collecting UCO in Hong Kong is strangely enough to be considered as a service, so they have to pay to get a UCO. However, they have to compete with other collectors, but those collectors export UCO to Europe instead of recycling the oil into something useful and provide it to the local market. So that's strange enough. However, the production of biofuels are still facing lots of problems waiting for the new generation to solve. So what kind of problems are we really facing when it comes to the production of biofuel? Like I would like of you to go to the website menti.com. I mentioned just before and type your comments there. And I will receive your comments. So you may now type in your answer. Okay, someone type contamination in biofuel. Unsustainable cost, economic cost, the usage. Yeah, those are the problems. It's said that the biofilm are costly as well because it involves many scientific processes. Yeah, pollution is also a problem when you consider to the production and the use of biofilm. Yeah, sustainability, cost, contamination, innovative, ethical issues. All of these are problems when considering biofuel. Yeah, that's right. One of the major concerns. Sorry. Okay, let's get back to our presentation. One of the major concerns of producing the biofuel is the problems of contamination. Among various contaminations, microbial contamination is the most challenging problem. Hence, our team is targeting the degradation of biofilm since biofilm is the source of microbial contamination. We are now moving to the second key idea, biofilm. So what is biofilm? Biofilm is a collective of one or more types of microorganisms that can grow on many different surfaces. Biofilms are made by both prokaryotes and eukaryotes. Microorganisms that form biofilms include bacteria, fungi, and protists. They appear in biofilms to allow microbial contaminations. Biofilm formation begins when free-floating microorganisms, such as bacteria, came in contact with an appropriate surface and begin to put down roots. The first step of attachment occurs when the microorganisms produce a gooey substance known as an extracellular polymeric substance, EPS. EPS are fundamental.
Sorry, Calvin.
Yes.
Our slide is not moving again. Maybe we let Kevin to help us to share screen.
Okay. Maybe we start from slide 11, Kevin. Slide 11, yeah. Oh, our technical problems. I'm really sorry for that, and we don't expect to have such a technical problem. Yeah. So maybe we have to wait for a while. Okay. Okay, Amanda, can you try moving the... Oh, okay. Let's continue our presentation from slide 11. Okay. When EPS formed, EPS are the fundamental constituents of biofilms, which enable the microorganisms in a biofilm to stick together. EPS are rich in high molecular weight polysaccharides, which is 10 to 30 kDa, and they have had a polymeric composition. And now Cindy is going to continue the presentation.
Hi, I'm Cindy. For microorganisms, living as a part of biofilm comes with certain advantages. For example, the slimy EPS covering can act as a protective barrier. It can help prevent dehydration and act as a shield against ultraviolet light. Also, harmful substances such as antimicrobials, bleach, or metals are either bound or neutralized when they come into contact with the EPS. However, from the perspective of biofilm production, when microbial biofilms are formed at oil or water interfaces in storage tanks containing dissolved biodiesel blanks, they cause a number of problems. For example, bacterial contaminants can inhibit ethanol production in biofilm fermentations and even result in stuck fermentations. Contaminants may persist in production facilities by forming recalcitrant biofilms, causing a degradation of fuel, and first, a lower efficiency in biofuel production. Hence, the formation of biofilms in biofuel increases the economic and environmental losses. Biofuel is biodegradable due to the presence of contaminations. It means that it is very likely to decompose if it is not sealed properly. Once biofuels are degraded, they lose their function to generate clean energy, which causes an economic loss to the society, and it takes capital to handle the waste biofuels. According to the Global Trends in Renewable Energy Investment 2020 project conducted by the UN, the world invests over $123 billion in developing biofuels. If we do not treat the problem of biofilm effectively and let it adversely affect the production efficiency of biofuel, we will be losing billions of investment and capital every year. The International Renewable Energy Agency foresees that advanced biofuels have a role to play in meeting the Paris Agreement goals. If the problem of biofilm is not well solved, we may not be able to limit the rise in average global temperatures to well below 2 degrees Celsius. Also, no country in modern history has reduced poverty without massively increasing energy use. Energy development is a key to economic development. The problem is that typically, countries must import oil from energy. leading to a negative trade balance, which can lead to inflation and exchange rate devaluation. Many countries, like Brazil, have experienced a negative trade balance because of the need to import oil. It is first essential for countries to seek local solutions. Therefore, if we could increase the efficiency of biofuel production by controlling contamination from biofilm, countries could have a lower dependence on unstable foreign trading. And now, Jackie is going to walk us through some major approaches to degrade biofilm.
Hi, I'm Jackie. We are moving to the next key idea, degradation. Designing a way to treat contamination from biofilm is the main problem we are facing now. Even with adding very toxic chemicals, it is not treatable. If you add too much chemicals, they will release into the air when burnt, which cause contamination to the environment. Various chemicals such as phenols, alcohols, heavy metals, and surfactants have been historically used for degradation for biofilms. However, due to their health hazards, these chemicals are not suggested to be used. Triclosan is one of the bisphenol compounds that have been used to degrade biofilms, but this chemical possesses several health hazards such as disruption of mitochondrial function and calcium signaling, and alteration of homeostasis and immunological parameters. Heavy metals such as mercury, silver, copper, zinc, and nickel, as well as chlorinated compounds, have been used for biofilm dispersal, but these chemicals are no longer in use due to their serious health hazards. Some research shows another approach to tackle the contamination of biofilm, which is to use antibiotics. However, it is said that antibiotics seems to be not effective for biofilm treatment, and in fact, it is expensive. As these cells have to be embedded in EPS, therefore, most antibiotics do not effectively penetrate the four-layer outer wall of biofilm formations. Moreover, antibiotic therapy given before biofilm has been adequately dissolved induce bacterial mutations, creating even more resistant phenotypes. The more we use antibiotics to treat biofilm-producing microorganisms, the stronger and more resistant they became. Successful treatment of biofilm-associated infections is troubled due to high antibiotic resistance in these bacterial communities. Therefore, we have to find another way to degrade biofilm, and Teresa will show us what it can be.
Hi, I'm Teresa. We now move on to another key idea, enzymes. The degradation of the biofilm matrix by biofilm matrix degrading enzymes such as DNase 1, dispersin B, and alpha amylase is so-called a better anti-biofilm strategy. Biofilm comprises the crammed bacterial population by extracellular matrix, ECM, which possesses bacterial secreted polymers such as EPS, extracellular DNA, eDNA, proteins, and amyloidogenic proteins. DNase 1, dispersin B, and alpha amylase degrade eDNA, biofilm, matrix, and EPS, respectively. They can inhibit the formation of biofilm and degrade the mature biofilms in various microbes, such as S. aureus, Vibrio cholera, and P. aeruginosa. Among those enzymes, two candidates are chosen due to its popularity and ability to degrade the biofilm components. The first one is dispersin B. It is a 40-kDa glycoside hydrolase produced by the periodontal pathogen. It releases adherent cells from a mature biofilm colony by disrupting biofilm formation. The enzyme catalyzes the hydrolysis of linear polymers of N-acetyl-D-glucosamine, NADG, found in the biofilm matrix. Dispersin B prevents the formation of at the premise biofilm, which suggests that biofilm-releasing enzymes can exhibit broad-spectrum activity and could be potential anti-biofilm agents. Currently, there's interest in dispersin B as a commercial anti-biofilm agent that could be combined with antibiotics for the treatment of bacterial infections. The second one is alpha amylase, which is our target enzyme. Because it is easier to obtain, if it is easier to get, its cost would be theoretically lower. It can be obtained from plants, animals, and microorganisms. Alpha amylase is widespread among living organisms. In the digestive system of humans and many other mammals, an alpha amylase called typin is produced by the salivary glands, whereas pancreatic amylase is secreted by the pancreas into the small intestine. But for the commercial application, alpha amylase is mainly derived from the genus Bacillus. The optimum pH of alpha amylase is 6.7 to 7. The amylase has a three-dimensional structure capable of binding to substrate and, by the action of highly specific catalytic groups, promotes the breakage of the glycoside links. Alpha amylase is an essential enzyme for alpha-glucan metabolism function to catalyze hydrolysis alpha-1 for glycosidic bonds in glycogen, starch, and some other related polysaccharides. There are tons of kinds of polysaccharides, but they all have one same property. They are polymers of monosaccharides through the connection of glycosidic bond. Therefore, alpha amylase is useful to break glycosidic bonds and then the polysaccharides. The enzyme weakens and prevents the matrix from encasing the cells within EPS and facilitates communication among them through biochemical signals as well as gene exchange. Then, the biofilm releases microbial cells from the surface, hence destroying the biofilm structure. Due to various limitations of chemicals and antibiotics used for biofilm removal, there's a need to replace chemicals with enzymes. And now Angel is going to show us what organisms can be engineered.
Hi, I'm Angel. We can now move forward to the final key idea, microorganisms. After determining to make use of enzymes, what organisms to be engineered is not a big issue. By doing research, we find that some organisms are feasible. 1. E. coli. The first one is the most common one, E. coli. Notice that the problem of biofilm also exists in water treatment. By looking at the method people use to treat water, we may get some idea. In water treatment, the iron bacteria and biofilm-producing bacteria grow quickly, where organic nutrients and mineral ions are accumulated under water cycles, causing corrosion of cooling towers. The cause of blocking and corrosion in cooling towers resulting from the colonization of microbes is huge. By constructing engineered E. coli, they design an integrated gene circuit which assembles sensing, cleaning rust, animating biofilm, and killing iron bacteria to solve this problem. Back to the problem which exists in biofilm, E. coli is also useful. By engineering an E. coli, they can swim to and destroy the biofilms to decontaminate the fuel tank and help preserve the biofilm in a better state. The project starts with using a gene called RER to detect the presence of C4HSL, which is a sensing cell of a bacterium found in most biofilms. Then the protein conducts the transcription, and the motor in E. coli will rotate in a straight path as expected. Finally, the engineered E. coli will produce chitinase and stenaldehyde to kill the fungi and bacteria. 2. Bacteriophages. The second one is bacteriophages, which literally means bacteria eater. They are viruses that target and infect specific bacteria. Following infection, the bacteriophage hijacks the bacterium cellular machinery to prevent it from producing bacterial components and instead forces the cell to produce viral components. Eventually, new bacteriophages assemble and burst out of the bacterium in a process called lysis. By using phages, you will be able to only target the hostile bacteria. Food industry uses bacteriophages on cheese to kill some bacteria to ensure the food safety. Bacteriophages is considered as a more cost-effective option. They infect and multiply in a bacteria starting from a small dosage. More importantly, they have no negative impact on human and animal cells. And now, Suki is going to tell us another microorganisms.
Hi, I'm Suki. We decided to use the third one, LDSB, which is a family member of the Lactobacillus species. There is also a helpful property which Lactobacillus species have. When they are under a low pH, for example, 3.5, they reproduce hydrogen peroxide. Hydrogen peroxide is used as a popular disinfector, which kills bacteria and is supposed to have a positive effect on the degradation of the biofilm. It secretes lactic acids that further biofilm degradation when hydrolyzed polysaccharides provide nutrients for LDSB. Lactobacillus species can be found naturally inside the human body, intestine, and vagina. They help to maintain the health of vagina by keeping a low pH around 3. Lactobacillus species are also used in food processing processes. So, they are considered least harmful to both the environment and people's health. Due to its usefulness in neutral fermentation processes, specifically in how it makes fermented food products out of cow's milk, it has great economic importance. So, it is as easy to get lactobacillus as it is to get E. coli. LDSB is the main bacterium used for the production of yogurt. LDSB not only can be the least harmful to both the environment and people's health, but also have a positive effect on the degradation of biofilm. As E. coli is too common to be used in the gene editing, we decide to make some challenges for us by choosing another less common bacteria as the target bacteria to be engineered. Therefore, E. coli is not our first choice in engineering. So why we are not choosing bacteriophages is that it is a virus. We do not have much knowledge in this editing a virus, so we decided not to put too much pressures on ourselves. To balance between innovation and feasibility, LDSB is the best choice. Also, LDSB is less harmful to the environment and humans' health, so it is more friendly for usage. We expected the biofilm produced in the presence of alpha amylase would be less robust than those in free amylase media. So Kelvin is now going to finish the following.
Here is our conclusion, and we are very interested in using engineered LDSB to degrade the biofilm with the help of alpha amylase. And we would like to degrade the polysaccharides of EPS in biofilm so as to weaken the structure and even destroy the biofilm. So here comes our Q&A question session. So Amanda, you can stop sharing your screen and I will take charge of it. Yeah, you may, yes. And all of you can go to menti.com and type your question there.
Or let's maybe open the Q&A section to
That's okay.
panel as well.
Okay.
I see Kenneth typed 4 questions in the chat box. So Kenneth.
Yeah, so basically the 4 questions is listed in the chat. So you can take a look, or I could also explain a bit. So first one, I've like kind of aware that there's a biofilm contamination and there's a current treatment, which is like a chemical methods. So like there are this group of chemicals are called the biocene, and what is the current limitation to it? And also second question is what's the major kind of bacteria that is contaminating? So is there like specific components in the biofilm, and does your enzyme could target those kind of biofilms like specific to those species? Third one is that if you're using a microorganism to treat the contamination, then the problem arises, does the microorganism also causes contamination itself? So can it propagate and live inside the biofilm and actually cause contamination itself? And how do you actually remove the original contamination if the if you're only proposing to degrade the biofilm but not to kill the bacteria, I mean the bacteria is still inside the fields, right? So how would you actually like remove all the bacteria? Yeah.
We may answer the first question. Okay, the current chemical additive which is biocin, and yeah, it's known that many chemicals are used to degrade biofilm for the current industry use, but it is said that they are harmful to the environment and to human because they have serious health hazard. For example, some we have mentioned about heavy metals are used and they are chlorinated compounds, and some chemical biocin has been used in the degradation of biofilm. We decide to use to degrade the biofilm biologically due to because it's more friendly to economic to environmental friendly to degrade the biofilm in a biological way but not chemical way, because chemical way is sometimes toxic to the environment or to the body, to the human. So the second question is the content our bacteria. Yes, and our enzymes, alpha amylase, inside the LDSB bacteria is weakening the structure of biofilm to maybe like it to make it decompose in the biofilm to or to prevent it from lowering the efficiency of producing the biofilm efficiency. So it is we found research and know that all biofilm have polysaccharides, of course, and most of all of the polysaccharides have a bond called, if I remember, it's called glycosylated bond, something like that. So all of the biofilm have this bond, and alpha amylase is responsible to break up the bond, so it can be weaken the structure, or even if we have time, or if we have make more research targeting how to even kill the biofilm, kill the bacteria in biofilm. Yeah. So the question number 3, our bacterium LDSB has a optimal pH value at low acidic environment, let's say 3 to 4, yes. And yeah, most of the alpha amylase have a optimum pH for 6 to 7, but there are some exceptions because there are many different bacterial alpha amylase or fungal alpha amylase. So actually we have found some examples of alpha amylase that work best in acidic environment, such as 3.4 or even lower. So yes, we can maybe we can got inspired from the John School team, and we may test which alpha amylase is the best, and yeah, test which is the best and choose to use which alpha amylase in our project. Yeah. So the number 4 question is about contamination. Yeah. This is our phase 1 because we like to weaken the structure of biofilm, but the bacteria inside we do not have so much effort to even kill them. Yeah, but once the biofilm structure is weakened and it have less negative impact to the efficiency of production of biofilm. So if we have to make further research and have further brainstorming, and we can even add some enzymes we have found different kinds of enzyme. For example, the DNase 1 and some certain protease from some bacteria species, and they can be responsible to degrade the bacteria inside the biofilm. So after we weakening the structure of biofilm, we may we may like to after weakening the structure, we may like to kill the bacteria as well. So Cindy in the chat box gave us some example of alpha amylase that optimum pH value is
Rainbow 6 share 因为 area F2
okay. Hannah asked that what is the role of bacteriophage in the project? It is our candidate in choosing which microorganism to be engineered, and we have E. coli, and bacteriophage and LDSP. And after comparing their advantages and disadvantages, we choose to use LDSP. So bacteriophage is like candidate waiting for us to select and which microorganisms to be engineered and up till now, our progress, we choose LDSP and alpha amylase, these two candidates to help us degrade the biofilm and weaken the structure. Yeah. So, any questions to help us do better?
Does anyone else in the panel or in
Okay.
the
If there's like nothing, I will no one else. I'd like to ask like another just. So basically, what we are seeing here is a kind of a paradox, right? So if your host strain could survive in biofilm, it is itself a contamination. But if your host cell is not able to survive really well in biofilm, it cannot produce the enzyme very efficiently. So how would you solve the problem and also the paradox?
Actually, there are some iGEM teams before to have used E. coli to produce alpha amylase and to weaken the biofilm, and they are successful in making E. coli to survive a long time in biofilm. So yeah, I think after we making more research, I believe we can make LDSP to survive in the biofilm as well by maybe looking at that iGEM team's method to let them E. coli to survive in biofilm. So yeah, LDSP and E. coli are the microorganisms can be used in releasing the alpha amylase, and we decide to have more challenges by not using the simple, the common E. coli and use LDS.
So basically, my point here is when you talk about how the E. coli can survive in biofilm, right, and also your host organism can survive in biofilm, won't it itself is a contamination? If you introduce this kind of remedy strain to treat contamination, but actually you're introducing contamination, how will you solve that?
The bacteria is kind of contamination, and that's right. Because, however, we can think in another angle. Biofilm, the contamination of microbial contamination of biofilm is bigger than the microbial contamination of our host bacteria. So after balancing, it seems that we introduce some little contamination to the biofilm to deal with a big contamination. It is quite good and quite meaningful and quite cost effective. And maybe we can find some methods to lower to make the contamination as little as possible. So we may find some researchers to see how can we weaken the LDSP without destroying the function of this bacteria. So yeah.
Okay. Thank you. So if we do not have any other questions or comment from our panel or from the audience, then thank you everyone for joining us in the morning section. So we will now take a short break before starting our afternoon section at 1:30 p.m. So please kindly join through the same Zoom link, and we hope to see you all there.
So please come back at 1:30 p.m. because we'll start off with another keynote lecture at 1:30. Yeah, thank you.
Okay, thank you. And beyond. Professor Wu is an assistant professor in the Division of Life Science, Department of Chemical and Biological Engineering in HKUST. She is also an associate director for the Center of Epigenomics Research. Professor Wu completed her bachelor's in bioengineering from University of California, Berkeley, before obtaining her master's and PhD from Stanford University in bioengineering. Currently working on single-cell genomics, microfluidics, and development of biotechnology, Professor Wu's goals are to help bridge gaps between biology and engineering. Her research focus has two major directions: investigating disease mechanisms using novel approaches such as single-cell transcriptomics and gene editing, and development of new technologies and tools to address experimental challenges in life science research. In today's talk, Professor Wu will discuss the ability to unlock the full potential of ongoing scientific research in the field of synthetic biology through their translation capabilities. She'll discuss some of the leading syn biotechnologies today and give case examples of their translation into the commercial space, as well as discuss the potential ethical pitfalls of such emerging technologies. So Professor Wu, please.
All right. Thank you both for your kind introduction. Let me share my slides. All right, so can everyone hear me and see my slides?
Yes.
Okay, great. So hi everyone. First of all, I'm really impressed that there are so many of you here on a Saturday afternoon when you could be outside enjoying the nice weather. But I'm very honored to have been invited to speak here. I know that some of the previous keynote speakers gave quite technical talks on synthetic biology. I kind of want to give a slightly different perspective. The reason is because my background is not only in doing scientific research, but I also, excuse me, have a little bit of experience on the commercial side, both in entrepreneurship, starting a company, and also worked briefly in management consulting. And so I think my view of synthetic biology as a new field that is full of innovation and a lot of commercial potential is really, I think, maybe a little bit different from some of the other speakers, and I wanted to talk about that. And also at HKUST now, one of the courses I teach is on ethics. And obviously with such new technologies, some of the ethical issues that arise are also very interesting to think about. So I want to draw awareness to that. Okay, so I'm going to start a little bit dry, just to give sort of like an overview of the entire field and sort of where it is right now and where I feel like it's going. So let's go back to basic definitions of what is synthetic biology. And I like this paragraph that describes it. It's the element that distinguishes synbio from traditional molecular and cellular biology is really the focus on the design and construction of core components, and that they need to be able to be modeled and understood, and also be tuned to meet specific performance criteria. And also, synbio involves the assembly of these smaller components into a large integrated system, specifically to solve specific problems. Just as engineers now design integrated circuits based on the known physical properties of materials, and then fabricate functioning circuits and entire processors with high reliability, this is also important, synthetic biologists will soon design and build engineered biological systems. So I think that from this you can see, I highlighted a couple of key components, and I think all of these components are very important to synthetic biology. And the way that I view the field right now is that I see people coming from different fields to address all these different areas in synthetic biology, and each of them see new innovations every day, which is very exciting. And I think the ultimate goal is this last part, which is, can we get to a point where we can design biological systems just like electrical engineers would design an integrated circuit in a very predictable, predictable and controllable way. And so if we then break that down into the 4 components of synthetic biology, I see the 4 components as first building models, one section, and then modifying and manipulating existing living systems, and biology as a technology. And then a corollary to all of these is being able to create the basic building blocks of biology to make synthetic biology more systematically engineerable. And I think people who have experience with the iGEM program will be very familiar with this part of it. So building models, what we mean is it's very similar to what Professor Huang just talked about, right? Understanding the physics and the math behind biological systems and using these fundamental principles to model biological systems to test and validate our understanding of them. As Feynman said, and Professor Huang quoted Feynman as well. As Feynman said, we cannot, we can't really say that we really understand something until we can replicate it, right? And so similarly, with synthetic biology, we really want to show that we can understand it by modeling and predicting biological behavior. Modifying and manipulating existing living systems. This is something that is very moving into a very exciting stage now. After we've understood these biological systems, we can extend them and manipulate them to create new things. And sometimes we can also view this as an extension of synthetic chemistry. Examples of this would be, can we make basic synthetic amino acids or modified proteins with new functions so that we can either reproduce or mimic natural ones, or even sometimes replace natural ones with the ones that we've created. And finally, biology as a technology. This is probably closest to our daily life. We are creating new biological systems for specific human-oriented needs. So this is what you sort of read about in the news. We create the engineered bacteria or yeast that makes fuel, or that digests plastics and help humans to achieve something that we want to do. And also at the very end here, this sounds very sci-fi, but one day we'll be creating completely new biological organisms as well. So these 4 components, I just wanted to give a quick overview of some of the interesting work that we've seen from the field, sort of touching on each of these areas. In terms of building models, one group at Stanford University, the Covert group, they've accomplished whole cell modeling in silico of the simplest sort of example of the cell that we know. And this is the mycobacterium, and it has the fewest number of genes for a bacteria, and they can model 100% of the genes in different sort of biological pathways in this bacteria. And so they've been able to show that using their in silico model, they can input different parameters into the model and get out the behavior of the cell. And then when they do the experiment, using these initial input parameters in the experiment, they get the appropriate biological output from the real cell culture. So I think this was really impressive because, as you all know, that all the processes inside a cell can be very complicated. And so to be able to accurately and correctly model all of those processes is not trivial at all. So this is a very interesting piece of work. If you're interested in knowing more details about it, I've included the title and also a YouTube link to a talk by Marcus himself on this work. Very, very interesting. Of course, as I mentioned, this is a very simple cell that we know. We're very far away from being able to model a mammalian cell, such as a human cell, at this level of detail. But it's a great proof of concept that we can build on. We meaning, scientists can build on to eventually achieve that whole cell model for mammalian cells. Okay, the next category was the modifying or manipulating existing living systems. And this paper just came out two or 3 days ago, which I found very exciting. Basically, someone from David Baker's group was able to computationally de novo design an entirely new protein, a transmembrane pore. And this is really impressive because, if you've studied biology at all, you know that proteins are very complex, right? They're made up of a linear chain of amino acids, but the way that they can fold up and form the actual protein structure is very complex. And it depends on all the different interactions that those atoms in the protein molecule can make with each other in 3D space. Not to mention, it's also affected by its surrounding environment. so the other proteins that are nearby, or whether it's in a cell membrane or in a buffer, are there other ions and things nearby? All of these things contribute to the behavior and the structure of the eventual protein product. And so being able to say, can we predict based on a chain of amino acids what that protein is going to look like and how it will function is extremely difficult. And this case really shows that using the newest computational technology, they can design a protein from scratch and show that this protein, when it's made in a cell, actually functions as a functional biological core. So this is super exciting. It just came out 3 days ago. I also included the link there if you're interested in learning more about that. Biology as a technology, there's been just a huge amount of innovation in this space. I want to mention 2 examples which have turned into commercial opportunities. The first one is synthetic biology used to re-engineer bacteria to create a drug, right? So you can see in this graph in the middle, we're seeing here, artemisinin is, as some of you may know, is a drug that treats malaria, right? And it's one of the few drugs that's very effective for malaria. But you can see that this is sort of the price of artemisinin. It fluctuates a lot, and this was in the past, right? So you know, from like the early 2000s, the price fluctuated a lot, and you can see that the yield is also sort of the demand, the demand for the drug is increasing, but the supply is not stable. And the reason for that is because artemisinin is traditionally derived from a plant, right? This will be highly dependent on things like the weather and whether there is any sort of natural disaster that will affect the supply of this drug. So it's highly desirable for humans to be able to synthetically make this drug, but it's very difficult to do that from a chemical synthesis perspective. So Jay Kiesling's group from UC Berkeley approached this problem from the synthetic biology perspective. And they thought, okay, can we transplant the pathways that create this drug from the plant and to put it into a bacteria or a yeast and see whether that it can be made inside bacteria or yeast. So they used a yeast, and by doing synthetic biology engineering of this yeast strain, over time they were able to produce artemisinin, or the substrate that makes artemisinin. They were able to increase the yield by 10-fold, I think it was. This looks more like 20-fold. And so based on this technology, they formed a company called Amyris, and they're able to now manufacture artemisinin as a drug using synthetically engineered yeast. Another more recent example is the creation of synthetic production of opioids in yeast. So opioids are the main component, or one of the largest classes of painkillers. And this is from Stanford University. So this is the researcher, Christina Smokey, who was conducting this research. And they were able to demonstrate the production of opioids in yeast as well. And so they also formed a company called Antheia. And their goal is to not only be able to create opioids in a sort of more cost-effective manner, but also by engineering them in a synthetic biology system that is controllable, they can also think about whether they can modify these opioid structures to make them less addictive or make them more effective, and so on and so forth. Obviously, because opioids are very addictive and can cause people to become drug addicts, so this particular innovation received a lot of discussion in the news because it was very controversial. So if you're interested in reading more about that, I included a link here as well. But and finally, I want to also point out this, which is the creation of a new organism from scratch. So J. Craig Venter Institute in the US, they basically redesigned a very simple bacteria by breaking down the genome of that original bacteria and then trying to predict which parts of the genome are essential for the bacterial survival and replication and which ones are non-essential. And after they removed all the quote-unquote unessential parts, they proved that their synthetically engineered bacteria can actually survive and reproduce with a much compacted genome, a much more concise genome. So I would say to call this creating something from scratch is maybe not completely accurate, but it is still a very difficult thing to do. And so they did that, and it's a feat of engineering. So if you're interested in this, there's also a great TED Talk by Craig Venter that you can look at. This is arguably the first synthetic life form that we have seen, which is a microorganism that's completely constructed using genome technology inside the lab. And finally, what you guys use all the time, I even put the iGEM logo here, different sort of gene circuits, right, that can enact different functions inside the cell, and then combining these different circuits to do different things and elicit different behavior, both on the temporal scale and also when you receive different signals inside a bacteria. And I would say this is sort of the fundamental that underlies a lot of this other technology we've been talking about, because without this, you wouldn't be able to do all of those previous things with control and with design. So I think all of these 4 components are very exciting areas of development in synthetic biology to sort of give everybody an overview of the cool things that are happening here. So many of you have seen this before. Nowadays, there's sort of been a formalized pipeline of synthetic biology, right? The build, test, learn, and redesign, and then start again sort of cycle. And I want to sort of put this in the, or put the applications that we're looking at in the context of this engineering pipeline. And this pipeline was really only made possible and feasible to iterate very quickly based on 2 major technological developments, okay? The first is DNA synthesis, is NGS, next generation sequencing, which allows us to read DNA very rapidly and at a low cost. Right? So being able to sequence DNA means that we can learn from the things that we've built. So if we've created a new bacteria, or if we've allowed them to evolve in culture, we need something as a readout to see what's happened to the genome, or how has the genome changed, or if there's a new bacteria that does a specific function, and we're interested in finding out which genes are responsible for that, you need a readout to tell you that. And so NGS has made this really cheap and easy to do. This is the first thing that allowed SynBio to really take off. The second thing is DNA synthesis at scale, which is being able to write DNA, right? So after we've read, the genes that perform a certain function, we have to recreate that or piece those things together to build our new organisms, right? The building part. And so to build it, you have to be able to create strands of DNA and then put them all together. And without DNA synthesis at scale, building would not be very efficient. And so then your pipeline would be slowed down, right? So now you've removed the bottleneck from this component and this component, and this pipeline has gotten really fast, and that's really caused SynBio to grow much more quickly as a field. But the next thing, the most recent thing, which has really changed the entire game, is genome editing, right? And genome editing is so important because this allows you to test very easily. What do I mean by that? Well, genome editing means once you've built your organism, you can modify it at specific locations to see what then happens, right? You can provide, it's almost like a stimulus to see what happens to that organism once it's changed. And so having all 3 of these in this pipeline really then lets you iterate this pipeline quickly and create new synthetic organisms at a rapid pace. So these 3 things all kind of came to fruition in the last decade or 2. And I want to talk more now about the genome editing part. Okay. So I don't know if any of the previous speakers have already gone into details of genome editing, but I always like to say that genome editing is as easy as putting an elephant in the fridge, right? So fundamentally, if you think about genome editing, how does one change the genome? Well, I say it's easy as putting an elephant in the fridge is that, when people ask you, how do you put an elephant in the fridge? Your answer should be, you open the door, you put the elephant in, and you close the door, right? Simple 3 steps. So editing the genome is also simple. You cut the DNA, and you remove the DNA that you don't want, like the deletion that you want. You might want to add in a new gene, right? But eventually the third part is that you join the broken pieces back together to make a whole genome again. Right? So it's very 3 simple steps, and DNA damage and repair happens in our body all the time, and our body has its own ways to repair the broken DNA. So actually, step 3 is quite straightforward. You just need to let the cell do its own job. So the difficult part then is really steps 1 and 2. Right, so okay, here I just give an introduction to DNA repair. There's 2 different methods. I'm not going to go into detail because it's a little bit hairy, but if you're interested in learning more, you can click on the YouTube links to take a look later. All right, so the hard part of genome editing is actually in step 1, which is cutting the DNA, right? How can we make the cut in the right place? Because it's useless if we just make random cuttings. I mean, sometimes it's useful. But now we want to design things, right? So we want to specifically cut things in a specific place. And so this is where engineered nucleases come in. There are engineered nucleases in a lot of different families that have a mechanism of locating and making the double-stranded cut in a specific location. So for most lay folk, if you talk about genome editing, they immediately think, oh yeah, it's that CRISPR thing that I've heard about in the news lately, and it's very new. But actually, genome editing has been around for a while, and there are lots of options available, even before CRISPR came along. So I'm not going to talk about all of these options today, but if you're interested, you can go online to learn more about them. Today, we will focus on the CRISPR-Cas system because it is a system that is the most easy to use. And so for those of you in iGEM, you've probably seen this before. This is the CRISPR-Cas system. Specifically, this picture is a model of the Cas9. And basically what happens is you have the Cas9 protein that recognizes the template or the target that it needs to cut based on the sequence of this guide RNA right here, right? So the guide RNA is like a GPS device. It tells the Cas9 where to go in the into the genomic DNA to make the double-stranded cut. As you can see here, based on the base complementarity, it finds the target DNA and it makes the cut. CRISPR was actually discovered in bacteria. It is the bacteria's immune system against viruses. So I don't know if all of you remember how your own immune systems works, but basically your immune system receives a stimulus from, let's say, a virus or a bacteria, and your immune cells will build a memory of what that bacteria or that virus looks like, right? It will remember what the bacteria's protein looks like. So those are like the evil proteins. So next time the bacteria protein appears in your blood system again, your immune cells will remember and attack it. And this is exactly what the CRISPR system is in bacteria. So the bacteria have to fight against viruses. And so in bacteria, what happens is that as the virus invades, the viral genome will be cut into small pieces in the bacteria and incorporated into the bacteria's own genome in this CRISPR locus. This is why it's called CRISPR, it's because of the existence of this CRISPR locus. So essentially what happened now is the bacteria has built a memory bank of viruses that it has seen in the past in this specific array, right? And the next time that the virus appears, the bacteria will make its own RNA from the viral sequence that it has remembered in the past. And this RNA then becomes the guide RNA, which allows the bacteria's Cas protein to target the viral DNA. So this guide RNA will complement with the viral DNA and then cut the viral DNA. And obviously now that the viral DNA is cut, it's destroyed, and the virus cannot infect the bacteria anymore, right? Okay. So that's how CRISPR was discovered in the bacteria. And there's an ingenuity of the people who discovered them realizing that this could be an incredible tool, right? And so there's a commercialization of this tool happening after it was discovered in bacteria. Like, let's say, if somebody discovered this immune system in bacteria, the initial discovery was, oh, it's just a natural phenomenon. But somebody had to be smart enough to think, wait a minute, if we take this and apply it to something else, it could become very, very useful commercially. And so these are the people who had that thought. So from the UC Berkeley side, we have Jennifer Doudna and Emmanuel Charpentier. They discovered parts of the CRISPR system, and they started companies around the technology. So you can see that all of these companies, I think, now have IPO'd or been acquired for quite a lot of money. And on the MIT side, you have Feng Zhang, who also discovered CRISPR in some way, and he formed Editas, which is also IPO. And there's lawsuits and stuff going on between there. But again, the realization of this as a useful and incredibly useful and amazing tool led to its commercialization. So once CRISPR became a tool rather than just a natural phenomenon of it's the immune system of bacteria, what kinds of applications have developed since then? So many of you have probably heard about CAR-T. CAR-T, it means the chimeric antigen receptor of the T cell. And so here you have the T cell receptor. It's responsible for recognizing things that the immune system needs to attack, right? It's responsible for recognizing whether this is part of a normal human cell or it's part of a invading species or it's a tumor cell. And CAR-T, the most incredible application of this is that it allows T cells to be revived to recognize tumors, again, in a patient's body. So the way it works and why CRISPR is important is because the T cell receptor, as I said, recognizes the enemy, right? And so in a lot of cancers, these cancers are able to keep growing in the human body because the host T cells have stopped recognizing that cancer as a bad thing. And so the T cell sort of is asleep. So if we can re-engineer or we can add a new T cell receptor into the T cell that is more aggressive and can recognize the tumor, then the T cell will be sort of woken up and will be able to remove the cancer on its own. And so CRISPR, the Cas system that allows us to make incredibly accurate DNA breaks in the genome at the right location, allowed people to edit the T cell receptor in a very easy and controllable manner. And this is why CAR-T was really made super feasible once the CRISPR-Cas system, genome editing system, was developed. You can see how much this has sort of changed the treatment of cancers by looking at how many CAR-T clinical trials are happening worldwide. So you can see here, China is by far the country with the most number of clinical trials around CAR-T, but all over the world, everybody is creating CAR-T therapies. And this explosion in the number of CAR-T therapies, I think, has been made possible partly because of the existence or the discovery of these new DNA editing tools. Another example of what CRISPR can do for us is as a therapeutic against viruses. And this is actually the company that I co-founded. It's called Agenavir. It's now been acquired by Vir Biotechnologies. So Agenavir doesn't exist anymore, but some of our intellectual property is still being developed. And so the way that we envisioned this technology is to use genome editing to target viruses that are secretly lying around inside the human cells and waiting to make cells sick again. So in the example of, for example, Burkitt's lymphoma, which you can see an example of here on the left, Burkitt's lymphoma is caused by EBV, Epstein-Barr virus, okay? So when Epstein-Barr virus enters the B cells, the virus will remain latent, but it integrates into the genome of the B cell, causing mutations and so on and so forth. So it can, as it replicates, that virus can become reactivated and it can cause even more mutations inside the cell. So eventually these cells then can become a tumor. But if we can remove the virus from the cell before it causes all these problems, there's a potential that you can avoid the cancer. And also, if we can remove the EBV in existing cancer cells, the cancer cell actually relies on the virus to keep proliferating. And so if we can remove the virus from these cells, the cancer will actually stop growing and die. So in this data, what you're seeing here is, you see here all this different guide RNAs that we're using in the CRISPR system to target the virus inside the cancer cell. And you can see that these curves show the growth of the cancer over time. So you can see if untreated, then the cancer will continue to proliferate over 6 days. But if we add the CRISPR system and the guide RNA targeting the EBV virus in the cell, then some of the guide RNA will lead to destruction of the EBV from the cells, and the cancer cells will stop growing and actually they will die over the 6 days. In the picture you can see here, these are untreated ones, and then over time you can see that the DNA of the cell gets destroyed and the cell dies. here's data from the experiments done in the company. You can see if we have a cancer that doesn't have EBV in it and we do the treatment, then the cells continue to survive. But if we treat a cancer cell that actually has the EBV inside, then the cancer cell will die upon the treatment. So you can see that CRISPR can also be used as a therapeutic against viruses. And another recent example of this, although using a different mechanism, is actually targeting COVID-19. So this is a very recent research paper from Stanford University from a professor named Stanley Chi. They developed a system called Pac-Man, which basically uses the CRISPR guide RNA pool to target the COVID-19 virus. And they propose that if we introduce this guide RNA pool into the lung cells before the virus even attacks, then the CRISPR guide RNA already in the cells will be able to attack the virus as soon as the virus appears, leading to these cells having almost a vaccine or an immunity against the virus. And so this is a new technology that is very interesting and being actively researched to see if it has potential to become a true tool against the COVID-19 virus. CRISPR can also be used as a diagnostic. So you can see here, because the guide RNA recognizes very specific sequences, you can imagine that, let's say, if we want to detect the COVID-19 virus, the SARS-CoV-2 virus, you can design a CRISPR system that would have a guide RNA that recognizes the SARS-CoV-2 and then generate a signal when the SARS-CoV-2 virus is present. So this is a schematic that shows that. And one of the companies developing this technology as a diagnostic is Sherlock Biosciences, again founded by Zhang Feng, one of the inventors of the CRISPR system. And Sherlock Biosciences recently received FDA emergency use authorization for their rapid diagnostic against SARS-CoV-2 using exactly this CRISPR technology. Interestingly, the other group from UC Berkeley who discovered CRISPR as well, Jennifer Donna, she started another company called Mammoth Biosciences, and they came up with almost the exact same technology at almost the same time, using CRISPR to target SARS-CoV-2 fragments. And they also developed a technology a diagnostic using this technology. As you can see, they've received NIH funding to develop their COVID-19 test, and they're also collaborating with GSK, a major pharmaceutical company, to develop a over-the-counter coronavirus test. So this will be a very interesting case to watch these two companies to see how it plays out in terms of the commercialization of these technologies. Furthermore, we can integrate this kind of CRISPR-Cas system into hardware as well. So you can see here in this example, what they've done is created a graphene-based field effect transistor, which is almost like an electronic chip. And you put the reaction for CRISPR-Cas on top of this sensor. And when it detects the right molecule, when the guide RNA binds with the right molecule and the right template and is cleaved, it sends a signal to the transistor, and the transistor can give an electronic kind of output as the readout. And so this has also been developed as a diagnostic platform, and you can imagine that this kind of electronic device could be very nice to use as a portable sort of out in the field diagnostic as well. Further developing the Cas family, there's also been Cas proteins that have been discovered to modify RNA instead of targeting DNA. And so it's also exciting to see where this might go and what sort of applications might arise from this, feature of the Cas proteins. Finally, direct sort of editing in humans. This is the latest CRISPR trial. It treats a disease called Leber congenital amaurosis, which is the first example of in vivo CRISPR treatment. In other words, this is actually a CRISPR-Cas system that has been injected into humans to try and fix a human disease. And this Leber congenital amaurosis is a form of blindness. This is undergoing clinical trial right now. It's very exciting to see where this will go as the first example of a human inside the body treatment using CRISPR. Okay, so just now I sort of gave you a very quick overview of sort of first generally, SynBio and what I feel like are the different areas of innovation that's happening in SynBio, and then sort of focusing in on the genome editing aspect that is so important to generating all these sort of synthetic biology applications. And then finally, to wrap up, I kind of want to talk about what are the sort of potential ethical pitfalls around this technology. And I think many of you have probably heard about some of them from, maybe your family members or just people in general or the media talking about the fears surrounding synthetic biology and genome editing. And so just very quickly, I would like you to think about what is your own view? Is it ethical to use genome editing on plants and crops? Or what about animals or humans? And I know that earlier you guys had a talk from another keynote speaker on sort of SynBio in agriculture. So maybe some of this was touched upon. When I previously gave a different version of this talk before, the audience that I asked this question to, most of them said that they thought it would be okay to do genome editing on plants and crops. And then, some of them said, yeah, I still think it's okay in animals, but in humans, almost everyone said no, okay, that they didn't think it was very ethically acceptable. And so I want to kind of go through these assumptions. So as the examples that I've mentioned before, we've already done a lot of genome editing, right? Obviously, in the lab, we edit mice and monkeys to study health and disease. In the case of producing the drugs in the yeast, we've edited microbes and yeast for production, also for biofuel production. And we've also edited, plants for agriculture and mosquitoes to be released into the biosphere. But I think the main distinction that needs to be made here is that there is a pretty big difference between editing in non-human species and editing in humans or human tissue. And within the category of human editing, we also think about editing ex vivo versus in vivo. So ex vivo meaning the CRISPR editing or whatever genome editing happens outside the body. So in the case of CAR T, for example, you would take a T cell out from the human body, edit it inside a dish, and then put it back in the human when it's ready, right? Whereas in the human, you might be injecting the drug directly into the human, just as the blindness trial I mentioned, and the editing actually happens inside the human. So this would be in vivo. But even in vivo editing can be classified as germline or somatic editing, right? So if you edit the germline, that means that you're changing the sperms and eggs of this individual. And this change that's made to the genome would be passed down to future generations. Whereas in the blindness trial, you're only editing the cells of the eyes, and that would not be passed down to future generations. And so I think all of these different categories have different potential risks and ethical pitfalls associated with them. And usually when people talk about genome editing, they don't make this kind of distinction very clearly. First of all, I think that non-human genome editing should be discussed more. We do a lot of editing in the lab on animals. Obviously, there are ethics associated with animal trials that need to be talked about. Is it humane? What can we learn from these trials, etc.? Even in the case of the production of painkillers in yeast, right? We need to maybe ask the question, if we make it easier to make painkillers, and these painkillers can be very addictive and cause people to suffer from addiction, how can we regulate this in an ethical manner such that synthetic biology is mostly used for good and not end up being used for bad things? With plants and agriculture, let's say if we create a soybean with higher yield and is more climate resistant, there are still ethical questions such as how do we promote equitable access, right? Is it only rich people who can afford to, rich countries that can afford to have this technology? Will we generate more inequality in the world, etc.? And also, do these new genome products pose dangers to the ecosystem as well? So that's another question. And mosquitoes being engineered to stop spreading diseases like malaria, again, is it practical and does it pose dangers to the ecosystem? So I think a lot of these questions are not discussed very actively today, and I think that needs to change. So one example is the gene drive that causes malaria. In a gene drive, for those of you that are not familiar, is using the CRISPR system, we generate a gene inside the mosquito that eventually gets passed down to all the offspring of that mosquito, okay? Even if that gene will cause the mosquito to die. Because usually if there's, by natural selection, if a gene causes a creature to die, it usually will not get passed to the next generation. And then eventually that gene will be lost in the population, like here. But in a gene drive case, the CRISPR-Cas system is used to aggressively promote the inheritance of that gene to the entire population, even if that gene eventually causes the organism to die. Okay, so this is a gene drive. Now, in Hong Kong, as a case study, in 2018, there were around 20 cases of dengue fever confirmed in Hong Kong. And health authorities were very alarmed because they didn't want an outbreak of dengue fever. And similar to malaria, dengue is spread by mosquitoes. So it's a virus rather than a parasite, but in severe cases, dengue can cause death. Okay, so there was some talk, should we use gene drive to eradicate the dengue carrying mosquitoes, right? Should we synthetically engineer a mosquito that will cause all the other mosquitoes to die off, and thus eliminating mosquitoes and therefore eliminating the way that this dengue fever is spread? And the question is this a good idea for Hong Kong? So as a case study, right, we can take a look, and this is associated with what I mean about the ethics of the situation. If we look at the number of dengue cases around the world, you can see that the United States has 51,000 cases in 2013. The largest, the countries that suffer the most from dengue are Brazil and India. So you can see Brazil has about 2 million cases of dengue every year, at a death rate of around, I think, 2 or 3%. That's quite a number of, deaths, close to 500,000 deaths, right? So this is quite high. In Brazil, because the dengue is such a big problem, they actually tested gene drives to fight dengue in 2016 in a few cities. Okay, and the cost for this gene drive to be implemented in Brazil is about $1 million or 8 million Hong Kong dollars for 2 years, okay? In Brazil, they observed no severe ecological effects so far, and the mosquito population in the city was reduced by over 60%. And the number of dengue cases was reduced by about 50%, okay? However, does this make sense? It's clearly effective, but does it make sense for Hong Kong? Well, if we look at the number of cases of dengue reported in Hong Kong, in 2013, it was only 103 cases, right? 103 cases. Comparable population-wise, in Singapore, there was about 15,000 cases reported in Singapore, and yet Singapore has not adopted this. So my point of showing you this, this case study is that, first, on the one hand, we need to consider whether this novel intervention is effective. And clearly, in the case of Brazil, it was. But the cost of this, this intervention is 8 million Hong Kong dollars. So compared to the number of cases, or rather the number of lives that it would save in Hong Kong, the cost is disproportionately high. And finally, there are other alternatives that we can use. So for example, removing the sites where mosquitoes may stay and produce their eggs. People can wear bug repellent, be more careful in the summer, so on and so forth, mosquito nets, et cetera, et cetera. Much more low-cost alternatives that could be implemented sort of more commonly to alleviate the situation. And so the purpose of me showing this case is that it's not always so simple to say, let's adopt a new technology that seems to be very effective. You still need to look at the potential risks, right? In this new technology, we don't know the answers of whether the mosquitoes might develop resistance, whether the gene drives might spread to the other species, or what if the gene drive mosquitoes escaped to other countries and cause some ecosystem changes there. What if the dengue, because it can no longer be spread by mosquitoes, it will adapt to be spread by other animals because you're applying a survival pressure on the virus, right? And so all of these factors have to be considered when you think about, is it ethical to do so? Because there are risks and there are benefits, right? So evaluating all of these things, my conclusion would be that gene drives is not a good solution for Hong Kong, but it might be a very good solution for Brazil because the number of lives that it saves there is huge. Okay, so after that example with non-human editing, let's go back to human genome editing, right? As I mentioned, genome editing itself is not new, okay? But being able to do genome editing in humans easily is new with CRISPR-Cas9. And one distinction that we need to make, again, is the heritable versus non-heritable genome editing. Okay, so in the body, we have these reproductive germline cells, the egg and the sperm. And these are the only ones, when edited, would pass any changes to the next generation. The rest of the somatic cells would not be passed, the genomic changes would not be passed to the next generation. And I think this is an important distinction. I think many people are sort of opposed to germline gene editing. They think, oh, you know, in the future we're going to have designer babies, and then, you know, what if there's a mistake? Then it will be passed down forever. But actually, in reality, this is the least of our concerns. Very, very few people want to do germline gene editing. And in fact, there are very, very few diseases that exist that can only be fixed with germline editing. Most of the applications that people think about in terms of disease treatment are accomplishable with somatic gene editing. For example, the CAR T therapy or other types of blood cell-based therapies can be done even outside the body. And so, again, most of the applications commercialized today are non-heritable genome editing to address a disease. And the ethical concerns of those are quite a bit lower. As I mentioned, the difference in risk between an ex vivo treatment and an in vivo treatment are also huge. In an ex vivo case like CAR T, you can even filter for the cells that you've synthetically created for bad cells. So for example, have we accidentally created a tumor cell, and that's bad. And so we can try to eliminate that before it becomes a problem and before putting it back into the human. Okay, in the interest of time, I'm just going to jump ahead to what are sort of the challenges of commercialization of synthetic biology technologies. And I think one of the difficulties is safety. After we looked at the ethical issues, is safety, right? And so safety means that we need to do a lot of clinical trials. And in the US, the FDA trials for a new therapeutic can take up to 10 to 20 years. Okay, and biotech entrepreneurship is fundamentally very different from other types of tech. I think in Hong Kong in particular, we hear a lot about sort of the e-commerce technology or e-commerce entrepreneurship, or even, you know, things like fintech entrepreneurship, which seem so fast and glamorous, and they raise money very quickly, and the products come out very quickly. This is not the case for biotech, especially SynBio, which is a new field with very few established regulatory guidelines. And so there are longer timelines, and the cost of developing such a technology is much higher. So I think so far, especially in Asia, a lack of recognition of the unique challenges and requirements, as well as the stakes and the risks in the medical therapeutics entrepreneurship can lead to big problems in terms of expectations in outcomes and returns, and results in sort of disasters like Theranos, right? Where you have, you know, fraudulent results and patients being harmed in the process. And this can be very, very bad for a new field, because once you have a sort of scandal that shows this is not safe and it has caused people to die, it can actually make investors and regulatory officials cautious about further investing in this field. So being researchers in synthetic biology and in genome editing, we need to be concerned about these issues and how certain cases can affect the entire field in the long run. So you can see here from this drug development timeline that once again, like drugs and therapeutics in bio take much longer than the other sorts of entrepreneurship that we hear about. As a comparison to a software startup, you can see that medical therapeutics, including SynBio, the typical first venture capital raise is 10 to 100-fold higher than that of software. We're looking at, you know, 20 to 100 million Hong Kong dollars compared to 1 to 10 million Hong Kong dollars. And the timeline is about 1 to 2 years for the first milestone, whereas for software or e-commerce, it could be months, just a few months, or 1 to 2 years for the launchable product. For SynBio, it could be 10 years before you finish clinical trials and sales, up to 10 years, I mean. In terms of talent and personnel, this is also very different. In software and so on, we often see, you know, people like Mark Zuckerberg types, right? Young CTOs, young CEOs, 20 to 30, they graduate freshly, like they fresh graduates, and they start companies and they do very well. But I think this is a different expectation in the medical space. You need at least 1 or 2 experts with very deep experience. And so a typical CEO or CTO will be older in the medical space, about 30 to 40 years old, simply because the regulatory and so on are just much more complicated and require a lot more experience. So that's not to say that if you're young, you should not think about starting companies, but it's very important to get experienced people on your team to help you with that experience. And finally, let's just take a look at the examples to illustrate that. So for example, CRISPR, Editas, and Intellia, you can see the venture funding for each of these is upwards of 100 million US dollars. And they IPO also very high. So Series A was 25 million for CRISPR Therapeutics, 40 million for Editas, and 15 million for Intellia. And if we compare these to sort of the conventional startups that we've heard about before, WhatsApp was 8 million, Dropbox 6 million, and GoGoVan 6.5 million. So really it's an order of magnitude difference. And I think this distinction needs to be better understood by the investment community and also sort of the startup community in general in Hong Kong and in Asia. All right, so I'm just going to skip ahead here and mention that, HKUST, we are very, very much invested in the synthetic biology space and innovation. Some of you may have seen the news from last year that we received a large gift to start a synthetic biology institute at HKUST. And so in context of this, I think HKUST is ready to do more in synthetic biology, and we hope to see a lot more innovation in that space, much of which will be contributed by students like you who are interested in doing iGEM and synthetic biology related projects. Okay, so I'm sorry to have gone on a little bit long. Thank you for giving me your time, and I'm happy to take any questions now if there's any time left.
Thank you, Professor Wu, for your highly enlightening session. If anyone has any questions, you can raise your hand and ask your question to the chat rooms. We'll unmute your audio if you want to directly communicate with Professor Wu. I think we have a few questions in the chat room. Yeah. So the first question is from Kenneth. So he wants to ask, do you agree that there is a more shift of focus as observed in SynBio field or in iGEM competition, that the research focus more shifted to real application instead of theoretical researches? And what is your opinion on any trend in synthetic biology?
Yeah, great question, Kenneth. So I think part of this is sort of everybody's excitement about the space and where it's going in terms of applications and creating commercial products. And so everybody's thinking about translation. How can we make real products out of this? Obviously, that's important, and it's always the most exciting part of an engineering space like SynBio. But I think, as Professor Huang Jiandong also mentioned, SynBio, and as illustrated by many of the examples that I gave, SynBio is such an interdisciplinary field. And it requires knowledge from many, many different areas. Like the modeling that I mentioned before, a lot of the things that we can do in terms of application would not be possible if you don't have that fundamental understanding. And so I guess your question is, do I agree with the focus? Well, I think there is a need for both aspects. And I think, iGEM being a sort of event that's catered toward students, I think it is more exciting for students to work on things that are more application focused. But I do think that research on the fundamental side will continue, and maybe there are other avenues in which to do that, so if that is your interest. But I do see people in the space focusing on different areas and pushing it forward from all directions. I hope that answers your question. It's just my opinion. Doesn't speak for anybody else. What is my opinion on the trend in synthetic biology? As I mentioned, I think the application areas are really exciting. Metabolic engineering currently is the big wave, right? The engineering of yeast or E. coli microbes to produce drugs, to produce replacement like synthetic meats, right? All of those are sort of based around engineering of microbes from a metabolic perspective to create products. And I think that's sort of the current big buzz. However, in the future, I think some of the associated infrastructure needs to catch up as well. So for example, what are the sort of methods that we can use to scale these up? What are the technologies that we can use to speed up that synthetic bio pipeline even more? I think, with any technology, you have things that are growing very quickly, but then it'll kind of stop or slow down because of bottlenecks from other areas, right? So you need that to then catch up again so that both can go up. So I think your question of fundamentals versus applications, both of those will be very exciting. Yeah, that's sort of my view. I can actually see the other questions too. Maybe I'll just, okay, so Teresa asks, do I envision a future where SynBio and biotech become as involved in day-to-day function as electrical devices are nowadays? And in what ways might this happen? I think, yeah, I think definitely we'll get to a point where day-to-day people in the average person will contact something related to SynBio in their day-to-day lives. Like, for example, those synthetic food products that I mentioned, right? Like the impossible meats or synthetic dairy that is created using yeast fermentation as well. Those things I think will become more and more ubiquitous. You know, people are super aware that climate change is a huge problem and we need to fix that. And so some of the ways that we change the way we grow food or create food help with that. And with that push, I think more of these products will become commonplace. The other things like sensors, I think that we probably have a longer way to go there because it is the marriage of a conventional hardware technology industry and bio, which has always been a little bit difficult, right? Making them compatible with each other. Those will require some shifts in the paradigm that we, manufacture these devices. It will also require some really revolutionary technologies that allow better integration between biology and hardware, I think. But I think eventually there will be sensors based on these microbes that you guys have designed in iGEM sometime, like sensing hard metals or sensing some toxins. I think those will eventually come to fruition and then they will be sort of everywhere in the smart city. Okay, the next question I got is from Phil. I want to ask, as you observe, how large is the portion of professors involved in startups in Hong Kong? That's a difficult question because it's a little bit broad. Your question is a little bit broad. Do you mean specifically involved in synthetic biology startups or in biotech startups or startups in general? Maybe biotech. I would say many of the professors are involved, but many of the companies they're involved in will not be in Hong Kong. And the reason is because Hong Kong currently lacks the infrastructure and the ecosystem to really do a full-on biotech startup. It's a little bit challenging. And so many of them, due to cost reasons or personnel reasons, they will be in like mainland China instead, or even in the US instead. So I know many of the professors here are involved. They're either advisors or they've co-founded companies, but they may not just not be active in Hong Kong. If you are interested, you should just ask. A lot of professors, they won't promote the fact that they're involved. You need to ask them, hey, are you involved in something? And then they'll tell you maybe a little bit about what they're working on.
Okay,
Oh, okay. He has a follow-up question asking, what are these professors going to be responsible in the startup? Good question. So professors sometimes are co-founders and sometimes they will be advisors. Many of the times what happens during the translation from academia to commercial product is, in the lab, we'll invent something, either like in engineering lab, we invent something that's engineered product, or we might find something new, like a fundamental piece of knowledge that's important, like in the case of CRISPR. And from that discovery, we want to say, how can we turn this into a product? And so the professor might say, okay, I'm going to be the co-founder, and because I want to be heavily involved in directing where this goes. And so the professor can be a co-founder, and then they will recruit, let's say, a PhD level scientist. They might recruit another CEO to the company, and they will sometimes play both a scientific advisor role as well as a more sort of CEO type of role. Whereas on the other hand, a lot of times what might also happen is the student, like the PhD student or the postdoc who worked on this project in the professor's lab will go out and be the co-founder, and then the professor will just be the advisor. So in terms of the technical aspects, they might give some advice on how to proceed further with the development of the technology, etc. So there's a wide spectrum of roles that the professor can play. Obviously, that's subject to regulations from the university on how involved we can be in an outside company as well. But yeah, knowledge support is one of them, sometimes a little bit of operation, sometimes just experience as well, and networking.
Okay, thank you, Professor Wu, once again for your valuable time. I think we have one more question.
Oh, one more. Do we have time though to take this question?
Maybe due to time constraint, this will be the last question.
Okay, so the question is, as indicated earlier, whole cell computational modeling has been complicated, especially in mammalian cells. What aspect of knowledge is most limiting our progress in this area? This is from Li Young. Okay, very, very good question. And again, I'm not an expert in this specific area. I just, read a lot very broadly. So again, this may not be entirely correct. My personal understanding is that the randomness in biological systems currently makes it really difficult to model mammalian cells. Mammalian cells have a lot more genes than a simple bacteria. And the mechanisms regulating the interaction of like different molecules inside the cell are more complex in a mammalian cell. If you think about your biology class, right, there are a ton of different types of ways that proteins can regulate each other, or small molecules can regulate proteins, et cetera, et cetera. And furthermore, in the mammalian cell, there's compartmentalization as compared to a simple bacteria, which doesn't have compartmentalization. So in a simple bacteria, you can think of it as like a bag of molecules just jiggling, like molecules in a bag just jiggling around. So when you model that, you can use some simple physics or biophysics and chemistry equations to kind of get a rough idea and then manage that randomness that happens there. But with a complex cell with structure and more complex regulatory mechanisms that may be more deterministic, that becomes a ton more variables that you have to consider. So it's not simply just computational power, it's also our understanding of those pathways and how they affect each other, and just too many things to model at one time. So I think that understanding of the basic interactions within a cell is very important. The other thing is the computational power. And incidentally, I just read an article on quantum computing recently, which talks about modeling small molecule chemicals and how quantum computing can be used to simulate interactions between atoms, right? And previously it was very difficult to do with conventional computation because the computational rate was insufficient to model that many interactions between atoms at a quantum level. I think potentially in the future, quantum computing could also expand to or be developed to a point that it could help manage a lot of these randomness that we observe in a biological environment as well. And so these technologies could marry and give us a better way to do computational modeling in biology as well. Of course, built on that fundamental understanding of the biology of the cell.
Okay. Thank you, Professor Wu, for your valuable time.
All right, it's been really fun. Thanks for inviting me.
Thank you. Okay. Our final keynote speaker for the afternoon session is Mr. Philip Boyne. So before we start, I'd like to apologize to Mr. Philip for the delay. So Mr. Philip will be leading the session on democratizing biotech. Mr. Boyne is the co-founder of the SynBio startup Bento Lab and was one of the team members of UCL 2013 iGEM team. In addition, he served as a mentor and a judge for the iGEM competition on several occasions. Bento Lab is a startup which aims to create tools for accessible biosciences. It is an all-in-one portable laboratory that combines a thermocycler, a high-speed centrifuge, and a gel electrophoresis setup so that anyone can get started with biosciences right away. The startup aims to allow scientists to carry their DNA sequencing labs with them wherever they go, by providing portable toolkits with essential tools which can fit in their hand luggage. Bento Lab was inspired from UCL 2013 iGEM team's project, where the team developed a safe, user-friendly, affordable biotech laboratory in a 13 by 11 inch box. The company started at iGEM and was deeply shaped by the ethos behind the iGEM community. Philip will talk about the early influences behind the Bento Lab, from iGEM to citizen science and DIY bio, and the lessons he has learned about entrepreneurship in biotechnology. So Mr. Philip, please.
Can you hear me now? Was I muted before?
Yeah, we can hear you now.
Sorry, it's still a bit early here. So, well, thank you for the invitation, and I'm going to try to keep it short and snappy, and maybe try to get you back on schedule a little bit. So I'm really really happy to be part of the symposium and to just talk to you a little bit about our journey from iGEM to entrepreneurship. And we started the company when we were quite young, just after iGEM. So I think that applies to some ideas more than others, as the previous speaker sort of mentioned, in terms of the difficulty of entrepreneurship that's unique to biotechnology. But anyway, I'd like to just start with, for a few minutes, talking a bit about what we do at iGEM, and then dive into our origin and the role iGEM played and the entrepreneurship lessons we've learned. And then hopefully we have some time for Q&A. I'm really looking forward to your thoughts and questions. So, so this is a picture of our Prime Minister, Boris Johnson. And he is, I think, probably recently visited a laboratory and getting a tour of their facility. And we saw this picture, and he looks a bit confused. He looks a bit less confident than he usually looks. And I think there's something about biotech that really makes people on the outside feel that it's not very accessible. It's sort of dangerous and complicated and off limits. And that's also reflected in the ethical questions and kind of the debates that we have that don't really progress much beyond the question of, is it okay or is it not okay, and don't really get into a lot of nuance. And we think that's a real problem, because I think we fundamentally feel that biotechnology has a lot of potential, but that in order to unlock that potential, we really need to make biotechnology a lot more widely accessible. And that doesn't mean that everybody will be a bioscientist, but it means that you know, it's a lot more ubiquitous in society, and it's not perceived quite as much as, you know, this is something that happens only if you have, you know, lots of years and years of training and the right access cards to the right laboratories. So our mission is to try to make biotechnology a little bit more accessible and democratized. And I think in a way that this mission is somewhat aligned with what iGEM stands for. I mean, people kind of forget, because iGEM's been so successful and so influential, that when it first was proposed, it was a pretty audacious idea and a bit sort of ludicrous almost, that a notion that a group of undergraduate students could make a small prototype bioengineering project happen in a few summer months. That was a pretty far-fetched notion. And in part, that was possible you know, because of what synthetic biology was designed to be, this idea that bringing the principles of engineering and abstraction to biology, that you could make a research project that would maybe before require a whole PhD accessible to a team of undergraduate students. And so iGEM's and synthetic biology has made bioengineering more accessible and brought it down, the complexity of it. And so we want to kind of continue that push. We're not targeting all of synthetic biology. We're just starting with sort of the DNA analysis workflow and the basic tools, but we want to make those also accessible, not just to biologists and researchers, but to anybody who wants to do that. So how do we do that? What do we make? So I'm getting these waiting room messages here. So we create tools that empower people to use biotechnology, regardless of who they are and where they are. And our main product is also called BentoLab, and that's a comprehensive biotech laboratory. It's a lab that fits in your backpack. Actually, this picture that you can see, that's my old backpack before it broke, unfortunately. And you know, you can sort of see there in the backpack is this device, and if in about 30 seconds you can take that BentoLab out and set it up, and then it looks like this, and you're ready to go, and and do research. Oh, what happened there? And actually, I've got one here behind me, so I can just show you quickly. It's it's kind of like a laptop-sized footprint. You've got a 32 well thermocycler here that you can fully program through the interface and save your programs, do heat block and touchdown PCR and whatever. You've got a centrifuge in the middle, and that's not just like a tiny tabletop slow centrifuge for quick bursts. It's actually goes, you can set the speed up to 8,000 G, or so you can actually do DNA extractions with it and kind of serious work. And then you've got a module here for electrophoresis. So you've got a blue light transilluminator. There's a gel box that fits neatly on top of this. You can see it in the picture there. The lid of the gel box is orange, so it already works as a filter. And then you've got the gel voltage output here. You can set different voltages, 50 to 100 volts. And so you can sort of almost see your bands hopefully very quickly appear and run down the gel. So it's a pretty comprehensive setup that weighs about 3 kilos. And if you've got this, you kind of, I mean, you will recognize that you've got a lot of tools to really get started. And then when you're done with your work and you're ready to go again, maybe you're on an expedition, you can just put it back in your bag in less than a minute. And yeah, then you're back on the road. And so customers really keep sending us pictures like this, or tell us these stories when we meet with them or talk to them. It's kind of like a typical experience that lots of BentoLab users have, that they're really excited to put the lab in a suitcase or pull it out of a backpack in a lab meeting or in a classroom. And it's something that really feels very cool. So even sometimes we travel with them and we go, we fly and we go through airport security back when we were still able to fly properly. And I often take maybe my laptop out of my backpack and then also my BentoLab and just explain to the security people what it is, such that they're not too alarmed. And that's always a kind of amusing moment that leads to some interesting conversations. So that's our main product, BentoLab. And then we have a number of additional products and services. So reagent kits for each step of the workflow for the DNA extraction, PCR analysis. And we also have a kit called the Biotech 101 kit. That's sort of like a chemistry kit for genetics. So that it's a teaching kit for a novice who has never done any biology necessarily, on how to use BentoLab, how to pipette, and then how to use all the different modules and apply them to a range of different projects. So from analyzing, extracting DNA from saliva and looking for different SNPs, or doing meat identification, plant extraction of DNA and identification. So lots of sort of world tour of different sample types and projects. And then we have on our website so the online resources, including all the protocols of that kit, with lots of very, very short videos, so that somebody can actually at home learn how to do biotech pretty much by themselves. That's anyway the idea. So I'll just give you showcase a couple of really great examples of where BentoLab is being used right now and where it's kind of uniquely useful. So this is a picture of a lab setup one of the users had in Sumatra. It's by Ineke Knot, who is a Dutch researcher doing her PhD at the University of Amsterdam. So she studies great apes like gorillas and orangutans. And she has a setup there with BentoLab to do all the DNA extraction, PCR, electrophoresis, and so on, because she's based there in a veterinarian clinic for these great apes. So it's a clinic, they don't have molecular biology equipment. And so she used that lab to do the to identify parasites, that nematodes in the feces of these great apes. And she also actually used the Oxford Nanopore MinION sequencer, which you might have heard of. It's this tiny sequencing device. And she did all the sample prep, the DNA extraction and sample prep for the Nanopore on the BentoLab as well, which is actually a really popular combination. We didn't design it that way because we didn't know about the Nanopore when we first built the BentoLab, but a lot of people use them together. You'd almost think we made the BentoLab for Nanopore prep. Anyway, I'm mentioning this also because her research has actually just been published a few months ago in an open access journal. And she goes into a lot of detail about the challenges and the potential of these portable genomic setups in the field and what you can do. And so you can find out more on our blog. We did an interview with her a couple of weeks ago, and there's a link to her paper, and I encourage you to check that out. And another case study that I thought might be really exciting, especially for you, is the idea of hands-on teaching and learning and field expeditions. So there's a range of universities now that are using Bentlab to really transform their hands-on molecular biology teaching. Kind of similar to how in iGEM, for me, it was super exciting that was my first experience being in a lab. So for example, these universities have a sort of a fleet of bento labs to give to students for individual learning. And this is a picture from Jocelyn Benitez Alfonso, and she and her colleague Elizabeth Duncan from the University of Leeds led an ecology field trip to Spain last year, together with 40 students. And one of the projects they looked at was tracking the health of bee populations. You can find out more about on our blog. The bees were healthy, but it was such a great week for us because we were in our office and we kept getting all this Twitter buzz and activity from the students and the lecturers sending pictures and videos. It was a real sense of sort of excitement about being able to carry out real work in the field. So for us, that's always kind of the real privilege of the work, getting that feedback, because we pour so much energy into making these devices. It's really great to get we kind of vicariously experience all these amazing projects that people do with them. Much more recently, as you can imagine, we've had also a lot of people using Bentlab to work from home. So we heard from a number of different researchers who already had access to a Bentlab for teaching or field work, and then who had sort of the foresight to maybe take the lab home with them in case of a quarantine, which proved for some people to be very useful. So we've seen pictures of lots of different home setups in garages and spare rooms where researchers then were able to continue to do their work. So I think that's really cool. I mean, I'm kind of thinking that it's kind of clear that lots of other industries have benefited from the productivity and creativity that you can unleash if you have laptops and small engineering gear and home art supplies. So we kind of hope that maybe we'll get a bit more of that kind of flexibility and creativity in biosciences if you're able to do more work from more different places. So this is a picture, a tweet we got pretty early on during the lockdown in the US by Sean Stice, who works at the University of Georgia. So he was able to continue doing research work on Bentlab from home. And that was a nice thing to see for us. And of course, it's also quite meaningful right now for us to hear when research groups are using Bentlab to investigate and prototype COVID diagnostics. We've heard of a range of different projects, including this one at the University of Peru. And a lot of these groups are trying out Bentlab for that because they mentioned that balance between ease of use, and it's a design that's really has been designed not to be intimidating to novice users, but it's still a lab that's uncompromising when it comes to being research grade. So this combination allows for the diagnostics that could really be maybe decentralized and on-site, because the devices and the workflow can be so simple that a technician with minimal training can carry it out. So that's a very quick snapshot of who we are. We're really passionate about our mission, and we love making biosciences easy to use and meaningful to lots of people. We've been kind of lucky that we've captured the imagination of lots of people in the SynBio community, in the biotech community, like George Church at Harvard Medical School. He was very kind to us in a Boston Globe interview, he was talking about the future of synthetic biology, and he said, there's this device called Bentolab, and it's kind of like an Apple II moment for biology. And maybe I'm not sure if you're too young, but if you know the history of the personal computer, it's kind of a big deal, that quote. The Apple II was the first mainstream computer that people could buy and use in their lives. So it really started the whole personal computer industry. And it's obviously huge praise and a lot of big burden on our shoulders. It's, for us, totally aspirational. We're not nowhere near that, but we're definitely working hard to try to have that sort of impact. So next, I'd like to go talk about iGEM and synthetic biology and the story of how Bentelab got started. So I came to London as an undergraduate to study computer science at University College London. I'm from Germany originally, if you can hear. And in my first year of being a computer science undergrad, I saw this picture, and that had a huge impact on me. You've probably maybe seen this. It's a biofilm that's been given the ability to retain an image. And it's actually one of the earliest iGEM projects from 2004 in Austin, Texas. And that picture really spoke to me as a young computer science student. Because apart from this being a really great science project, the iGEM team also did an amazing job communicating. And they chose as one of the images that they exposed onto the biofilm, those two words, hello world. And for a programmer, those are kind of magical, special words. It kind of means I've programmed this. It's the first words that you put into a computer program. So as soon as I saw that, I kind of knew that, that this is where I wanted to be. I had this really strong sense that something important was happening here and that I wanted to be a part of that. So I jumped right into as much synthetic biology as I could. I joined the university's iGEM team. And during my time as a student at UCL, I've been involved in that team every year, from leading and facilitating teams, being part of the team, and actually in the end also judging at the competition. And I also founded SymBioSoc, a sort of student society in London, to help build the infrastructure and nurture talent for iGEM. So I was still studying computer science, but I pivoted my degree towards synthetic biology as much as possible. So my thesis ended up being sort of a shared project between computer science and biochemical engineering and biosciences. So actually I ended up having a desk in the systems and synthetic biology group, although I was an undergrad student writing my thesis. And my master thesis ended up being a sort of exploration of applying software engineering methods to the design of genetic circuits. And I had built some working prototype programming languages for designing genetic circuits using those approaches. So that was a lot of fun. I was super hooked. And during my time as one of the leaders of the iGEM team, one of the things I was trying to do was find a really impactful human practices project. I think it's called Practices and Policies now. It used to be called Human Practices. So, figuring out how to integrate your project in, make a societal impact. And I'd heard about this movement called DIY Bio, which you've probably heard of. It was kind of quite new and nascent and obscure back then. Do-it-yourself biology, biohacking. It's basically where the intersection of the maker movement, so people who are interested in 3D printing, making devices, hacking culture, and synthetic biology. So people with engineering and computing backgrounds often, whose imagination was also being captured by the promise of synthetic biology, kind of like mine had been. So I thought it might be a really good opportunity to try to facilitate a collaboration between our student iGEM team and this group of amateur biohackers that was small but active in London. And at that time, it was totally impossible for non-university entities to participate in iGEM. So that's changed in the years since then, but our project kind of became an exploration of what it would take for amateurs to go through the whole iGEM workflow outside of a university lab, and if that could be meaningful and a great addition to the competition. So, we worked with this group and we facilitated that work. They identified an interesting gene that they were interested in. We facilitated primer design workshops and they designed primers. We built equipment there to do all the steps of the work, like the PCR, the incubation and so on. We built this shaker incubator in a fridge box made out of cardboard and moving parts that we picked out of the hackerspace. And we ran the experiments there. I mean, I should say they ran the experiments there, and we just took the pictures. And finally, we helped them submit a part to the parts registry. So it was really, the iGEM team helped facilitate that, but it was really the biohacker group that did the work, built the equipment, made the decisions, and yeah, it's we called that the public biobrick. It's not the most useful biobrick in the parts registry, but it was kind of a performance and kind of a proof of principle that, you know, the parts registry could be, that you know, non-academic groups could maybe make use of it and contribute back to it. And on that project, on that iGEM team, I also met Bethan Wolfenden, who became a friend, and we quickly realized that we had sort of similar interests and worked really well together. And she eventually became also the co-founder of Bentolab. So actually, the two of us were really hooked and interested in this DIY bio community, and so we kept. We kind of went on a world tour exploring different DIY bio spaces and talking to the people running them and asking them, you know, what their challenges were, what they were interested in, because we just felt that there was something really powerful about the notion of getting more people and not just professionals into synthetic biology. And we eventually sort of started to crystallize this idea that, you know, this is what happened in computation, that in the 50s, a computer really was a laboratory. It was a room that professional people worked in, and these people were highly trained, just like lab scientists are these days. But then obviously we know what happened there. You know, we started having personal computers in the 80s, and that changed not just how much a computer cost and what it looked like, but also who actually was able to use one. So you didn't have to have a PhD in computer science to use a mouse and a keyboard and write hello on a screen, or maybe use it for your hobby project. So that really changed the ubiquity and the usefulness of computers. And then of course, you know, nowadays computers are in everything, and you can get, you know, $10, $20 Raspberry Pis that you can learn how to program with and build projects on top of. So we had this idea that, you know, maybe we could, and in the beginning it was almost like a design fiction, an idea that maybe we could build a laboratory that was like that personal computer in the middle. And we called that Darwin Toolbox. And we used iGEM as a catalyst. I'm a big believer in using sort of a tight deadline to try to come up with something quickly. So we assembled a team of students and we built a prototype and we entered it in the iGEM entrepreneurial competition. At that time, that was a special track within iGEM. And I just remember the Jamboree that was in 2013, where we had this little stand and we had that little box. It was just I mean, it didn't work. It was just a sort of make-believe arrangement of a centrifuge, PCR, and gel combination in this metal case. But we had such a great time during that Jamboree because we could really sense that it had captured people's imagination. I remember on the first day, Tom Knight, you know, one of the grandfathers of synthetic biology, sort of walking past our stand and then having a look at it and having a conversation with us. And he was clearly quite enthusiastic about this. And then the next day he came with a bunch of his friends to show it. And so we felt that we were onto something that was interesting. And so we were students at the time. So, you know, this was just a hobby project, but we kept kind of building and iterating on the idea and trying to make this object look more refined. And we gave it to labs to sort of imagine how they would use one, and eventually ended up with this model that we made 20 of by hand in our workshop, and then loaned out to different beta testers. That's what we called them. So we found that actually people found them very useful. And that's when we decided to maybe go ahead with this idea, even though we were just out of universities and didn't know anything about entrepreneurship and were horribly inexperienced. We thought, okay, it's now or never, let's try this. And we launched it as a campaign on Kickstarter initially. And we got for a biotech, consumer biotech project, a very high response. And yeah, we've been kind of running ever since. It took us quite a while to build the manufacturing capabilities and to kind of grow up and learn that we're now a company and not a student-run hobby project. You really learn the nitty-gritty of operations, but we persevered, and we've had a lot of really great responses, not just because people actually have the labs now and use them, as you can see in the case studies, and it's really making a difference to them, but also it's just like Tom Knight at that Jamboree or George Church, it's really managed to capture people's imagination. So it's sort of something that people feel like they can imagine what the future of really democratized and widely accessible biotechnology might look like. And that's been really fun and to be a part of that conversation. So to finish off with, I just wanted to give you a couple of thoughts and things that I learned from iGEM that's been really super useful on our entrepreneurship journey, and then we can dive straight into Q&A. So for many years, actually, one of my jobs on the UCL iGEM team and community was to recruit students to our iGEM teams and to pitch iGEM to potential candidates. And one of the phrases I remember that I really used very often to describe iGEM was that it's a bit like a mini summer startup. It's obviously temporary. It lasts for a short period of time during the summer, and then it's often over. But during that time, it really feels like you're part of a super high-paced biotech startup, because it shares many of the characteristics, and that's why I think you actually learn a lot about entrepreneurship and being in a startup from iGEM. And these are, I've just written 3 very simple ideas and principles that I thought might be useful to talk about and that might be relevant to you. And they sound super trivial and super simple, but that doesn't mean that they're not really key and they're simple, but they're not easy. So one of the principles is the idea of ship or die. iGEM does this really clearly because you have a deadline. You know that by some point, by the deadline, you have to submit your parts to the registry. You have to upload your data to the wiki. And I think that really focuses your attention on figuring out what is something that we can build in that time that we can ship, because something that's only half done and it's not shipped might as well be, it's basically nothing. Something is only actually creating value if it's at the point where you can show it to somebody and demo it to somebody or ship it to somebody. So that applies in startups, obviously, and that doesn't necessarily have to be a finished product, but it might be in a biotech startup, the next point where you can show an investor the progress. But yeah, it's really a mindset of shipping and delivering value early, and not sort of keeping tinkering and missing deadline after deadline after deadline. And of course, iGEM teams often have really big visions that will take 10 or 20 years. In our iGEM team, we had in 1 year we had the notion that we would use microbes to build an artificial island in the ocean, collecting all the microplastics that exist in the South Pacific and building a kind of sci-fi island. That's obviously not something that we actually did that summer, and we're still waiting. But we actually had a great, sorry, this is a tangent, but we had a great little outreach program where we had a fake Google Maps website where people could already buy real estate on our synthetic biology created island. So that's actually how we did crowdfunding for that project. Anyway, so you have obviously a big focus, a big vision, but then you have to really figure out what's the minimal viable proof of principle that we can construct over that short period of time, and that we can ship, and then we can build on top of that in the next iteration. And so that's a really crucial thing to apply. The second thing I thought I'd write down is don't hesitate. You have to make decisions and you have to own them. And of course, you shouldn't make decisions on a whim. You should think about them deeply and you should look at the data. But then I think very often people can overanalyze, and that leads obviously to this idea of analysis paralysis. And you ask 10 advisors and you get 20 different ideas. And at the end, you still have to, at some point, sit down and trust your instinct and make a decision. And very often, I think people take too long. They actually already know what the decision has to be, but then they still keep talking to other people and try to get more validation that it's really the right decision. And I think what people underestimate is the delay and the cost in not making the decision earlier and going ahead and actually learning something. And if it turns out that the decision was wrong, you're going to find that out much earlier if you actually go out and do some of the work, because that's when you actually learn more. And I mean, how long is an iGEM project? Maybe 3 months or so 90 days. And 90, and that's an incredibly short amount of time. So you really have to sort of not hesitate, but you have to make a smart decision and own it and take responsibility for it and then go for it. Because 90 individual days are very short. But if you have them in a sequence, I'm always amazed at how much you can actually get done if you're focused, if you make decisions quickly, and then just build on your progress every day. And suddenly 90 individual days chain up and you get sort of compound interest for your effort, and you build this momentum. And after 3 months, you can't believe how much progress you've made because you've been focused. If you've got 90 days, but you do something different every day, you won't get much done. And lastly, resilience is absolutely everything. Both in all of our iGEM projects, there have been days and maybe weeks where it felt like nothing was working, and we all felt like giving up. Or and in Bento Lab, it's certainly been like that. Sometimes months where it just felt that we were definitely going to fail, and then sometimes you have to still give presentations like this, and it really takes a lot of takes a toll on you psychologically. And most rational people would give up and not do that. But it's absolutely essential, if you want to do something hard, that you power through those, and that if you feel like there's no options and failure is inevitable, that you still show up for the work, you try to create new options, and you're resilient. And that's an absolutely key thing. And it sounds simple, but that's where most people fail. So iGEM has definitely taught me all those lessons, and I'm convinced that the experience that you're having in iGEM will serve you really well, whether you're doing research, which certainly has this aspect as well, or whether you decide to become an entrepreneur. So that's it. I just wanted to share this picture because I really love it. This is from a workshop, Bento Lab. Well, it's not our workshop, from a workshop that used Bento Lab in Japan near Hiroshima last year, called Interlab Camp. And it was this great workshop with 20 or 30 different designers and students who over 3 days learned from 0 to I don't want to say 100, but from absolutely no background, learning how to pipette and learning how to do research and learning to do sequencing and all that stuff. And they extracted DNA from different vegetables and contributed and created new sequencing data. And they used Bento Lab as part of that. And at the end of the workshop, and this is what I love so much, they all felt that they wanted to sign the Bento Lab kind of like a rock star guitar or like a cast or something. And to me, I was there. My name is somewhere over the logo. It was a really special moment because I felt that's to me symbolizes that people really felt that this was a lab that they could own and use and personalize, and they weren't intimidated by it at all. They just felt that, yeah, this is like my MacBook full of stickers. This is my lab, and so I find this really inspires me a lot, that picture. Lastly, I have one request, which is we do follow us on social media. We just started a YouTube channel, and we're interviewing one of our users every week from lots of different areas, and we could still use some more subscribers. So it would be great if I check later and we have a few more subscribers on our YouTube channel at Bentolab. Anyway, I hope that was interesting, and I hope you've got some great questions for me. I really want to hear from you, actually. Thank you.
Yeah, cool. Thank you for your engaging and inspiring sharing. So if you have any questions, you can just leave a question in your chat box, or you can also raise your hand so we can unmute you and you can ask your question out loud. And maybe, yeah, we have some comments in the chat room.
Yeah, I'm just catching up on them, actually. Let me, that's great.
So, yeah, we're rushing to follow Bentolab on IG. So, Jackie pointed out that like the idea of developing a portable ultra-low temperature fridge, do you think it's possible?
Well, so I'm not a physicist, so I'm not 100% sure. I remember actually many years ago seeing a prototype for something like that. So I mean, obviously that would be a super useful thing. I think for us, when we started, Bentolab, we were super steeped in synthetic biology, and we actually were much more focused on just synthetic biology as a market. So we had definitely a lot of ideas of adding more and more tools to the lab, and that's still something that's sort of in the back of our minds. But we also realized that to do something, we really had to focus on a few tools. So that's why we sort of decided to just stick with that mix of tools, because it's actually so useful in many different devices and in many different workflows. And the problem is when you think what's the next thing I'm going to add, it's not really clear what the next best thing is. It really depends on the application. So we're definitely thinking about what we might add, and I know that there are some portable fridges that are, I think, particularly for moving medicines that need to be kept cool to decentralized areas. But yeah, for us, I think we're trying to become, if anything, more focused in terms of what we build. Sorry, I'm shaking the screen here. Yeah.
Okay. So we also have a question from Jasmine. So Jasmine said, I suppose it's sold at a much cheaper price than conventional lab equipment to make it more accessible. So how do you reduce the production cost while not offsetting efficacy of the equipment?
Yeah. So the first, I mean, we had to actually find a quite difficult balance between making it affordable, but we also actually really quickly realized that if we made it too cheap, then people think it can't work. So we had to sort of find this balance, because as you've seen in the case studies, a lot of our customers are researchers, that we actually found that if you make it much cheaper, people don't take it seriously, and also, of course, you have to sell a lot more to make money, and we need to obviously invest in future R&D. So it's actually, if we have to sell 10 labs, then at a much cheaper price, it's harder for us to invest in research. So we had to find that balance between making it not too expensive, because we did want to bring the price down substantially. We want to enable more people, but then also not make it too cheap for it to be not taken seriously. In terms of how we achieved our cost, it's not, the production price is not as cheap as we wanted it to be initially, but we're still getting there. We're still making improvements to it. But it's really because we just built it from the ground up. So we didn't work initially with suppliers that supply medical equipment. We sourced all of our own components, and we were very strategic in terms of where we put our money and where we cut costs. So we didn't compromise in terms of the temperature probes in the lab. They're really expensive, high grade, same stuff that you get in regular, very expensive equipment, which is different to most of the sort of open sourced DIY biohacker devices. But then we also realized, well, if we bring the size down, if we use more consumer electronic suppliers for certain things, so we can bring the cost down. But yeah, I mean, we still definitely want to figure out how we can make sort of more entry-level models eventually. Please do consider selling your bento to Hong Kong. I think, you know, I'm trying to. Obviously, I can't mention any customers who I haven't cleared that with, but I know we have a few bento labs in institutions in Hong Kong. So and there are a few. It's really exciting actually when teachers. It's usually the teachers or actually students who convince their teachers to say, hey, we need to have an after-school class. You know, there's all those after-school robotics classes, and we're seeing a lot more students saying, well, why don't we have an after-school genetics class? Let's get this. So it's definitely not too expensive for a school to get a few and start a club like that.
So I have a question. So it was really inspiring to hear how you went from iGEM project to a company. So what I wanted to ask is, what is your plan for the future, like for Bento Lab? Like,
well, as I kind of mentioned, it took us a long time to really become a proper company, and I don't mean, we obviously were legally a company, and and all of that, but I think in terms of the mindset that we brought to it. So that's been something that transition we really made only in the last 2 years, where we were less just obsessed with the mission and the devices, and actually thinking, well, we have to build all the boring stuff, and actually starting to find the boring stuff like customer support and answering emails reliably every day and having somebody who does that and who's responsible for that, and shipping and just all of that stuff that you don't really think about when you just want to build the technology. So that's what we spent the last 2 years really focused on, just kind of becoming operationally excellent. And now we're actually just about to really think about where we want to be in 18 months. So I don't quite know yet, but we're definitely thinking about how we can sort of take the next step towards that mission. Yeah, I don't. We've got a lot of different ideas, and as I said, I think it's really hard. Instinctually, I always want to say yes to everything because I'm really excited by all these different directions. But of course, if you say yes to everything, you can't get anything done. So it's the hardest thing that we'll do in the next 3 months is from the hundreds of ideas that we have, only choose one and go with that. So yeah, that's what is in the future for me is very hard decisions.
So let's get the last question from Derek. Derek asks, will there be a license given to the licensed users after the tutorial section considering safety issue?
So I'm gonna have to interpret that a little bit in terms of what you mean by license. I guess when you say a safety issue, you sort of mean like I guess I could interpret that in two different ways. Either like a certificate, so that when you go through the tutorial, you get a sort of certificate that says you've done this. I mean, we can't give a license in the sense like a driver's license or like a health and safety license, obviously, because number one, we ship to lots of different countries, and yeah, I mean, we definitely take a lot of care, and iGEM has taught us this as well, in the design of our products, both the hardware and especially the knowledge resources and the wetware, that we don't just want people to kind of learn the workflow as a kind of cooking step. We want people to think about the human practices and the ethics and the responsible innovation around if they become a biotechnology practitioner. In terms of, and so definitely how to operate the stuff safely, how to dispose of things safely. I mean, obviously the reagents that we choose for the kit are designed with safety in mind. So, we don't have like ethylium bromide or anything like that. We try to find the right balance between something that's useful and performs well and something that's safe in a home context. Safe for somebody who knows that, it's still a scientific tool. And Yeah, this was also one of the reasons why we initially targeted more DNA analysis workflows as opposed to just genetic engineering workflows, because of course, once you start doing recombinant DNA, the regulation rightly becomes a lot tougher, and also a lot more different for different regulations. So here in the UK and Europe, it's very different to the States, and it's very different to Japan, where we have a big market. And I don't quite know what the regulation is in Hong Kong, but whereas for DNA analysis workflows, there's actually a lot of the stuff you can do without regulatory concerns. There are some rules in terms of, like, obviously looking for human, so doing diagnostics and stuff, but purely for research, just extracting DNA and looking and doing DNA barcoding type activities, that's usually something that's fine in most regions. So I don't know if that answered your question, but yeah.
Okay, cool. So thank you, Mr. Boeing, very much for valuable insight and sharing. Right.
Well, thank you for having me. It was great. And if you have any more questions, you can find us on Twitter, and you can email me at phil@bento.bio. So yeah, thanks for the time, and I hope you have a blast doing iGEM.
Thank you.
Thank you. We will now have the iGEM Hong Kong undergraduate team participants presenting their projects. Each team will have approximately 20 minutes for the presentations, followed by Q&A and feedback from the panel advisors and ex-iGEM members. Same as the morning section, we will give you notice at the 20 and 30-minute time points. The panel for the afternoon session consists of Kenneth, Tim Tam, Simon Shiu, Mark Rousseau, Hannah Liu, and Felicity Ling. The first presentation will be from the HTU team, who are working on the project maintaining a constant ratio of different phenotypes of bacteria in a mixed population with recombinase gene circuits. Synthetic circuits have the utmost importance in industrial bioproduction, environmental mediations, and even cancer treatment. However, current synthetic circuits are metabolically expensive to run and readily, especially if it involves more levels of control, including feedback loops or control of protein expression or cell phenotypes. Through their project, the HTU team proposes a synthetic orthogonal tyrosine recombinase-based gene circuit that is able to automatically regulate control without feedback loops or other metabolically expensive regulatory elements like Curum sensing module, kill switch, or toxins. On behalf of the HTU team, Stephanie, Eric, and John will now be presenting their project.
Hi there. Good afternoon, everyone. We're the Hong Kong iGEM team. Thank you so much for introducing us. And today we'll be talking to you guys about our project to build a gene regulatory network for the ratiometric control of bacterial populations. So by drawing inspiration from these recombinase-based systems, our team hopes to propose a new solution that can be used to combat the issue of microevolution in cellular cultures. So in the past decades, synthetic biology has expanded rapidly, and it's been applied to study and, in a way, control life as we know it. Scientists have designed, built, and introduced novel and increasingly complex genetic circuits into these single-celled organisms, and this has helped harness these simple organic life forms to fulfill functions that nature may have never really planned for them to perform. And so when we look back on what synthetic biology has achieved over the past decades, we see something that's very tightly regulated, and it's a reductionist framework that we're trying to engineer these target organisms into something that's more predictable, something that's more reliable, more like an algorithm in a machine. So with this kind of school of thought, chaos has absolutely no place in our science. But as biologists, we know that life is, well, anything but predictable. Sometimes our built synthetic systems just don't work. And sometimes it's because we overlook something in our experimental design. And sometimes it's just a tiny genetic mutation that just snowballs into this gigantic issue for our projects. So what I mean is like when the frequency of a gene within a bacterial population, if it changes due to selective pressures, well, this is microevolution. And this is an idea that we're very familiar with, counting with the terms of natural selection and whatnot. And in particular, microevolution in E. coli, one of our model organisms, has been extensively studied with the LTEE, the long-term evolution experiment that was conducted by Dr. Linsky since 1988, where he has been studying how 12 originally identical E. coli populations have slowly diverged and accumulated different genotypic and phenotypic characteristics over the years. So here on the right, you can see how mutated cells, the ones that we've highlighted in red in the flask, which maybe in that kind of environment may be genetically more fit, and they slowly gain dominance and are driving this ratiometric change of alleles within the population. Now imagine this happening over tens of thousands of generations and implicating a whole bunch of different mutations. This becomes a headache for synthetic biology because what we do when we design these systems, we want them to work exactly the way we do. We do want it to be. And these small kind of like changes can threaten to derail our designs and these meticulously ordered systems, leading to perhaps gradual losses of functions or the piling on of excessive metabolic burden that would render our synthetic organisms unsustainable. So clearly, in order to improve and optimize what synthetic biology has to offer, we first need a solution to combat microevolution. The ideal product in mind would be able to stabilize and prolong the viability of whatever kind of gene circuits we wish to introduce into our cells. Ideally, it'd be self-regulating, not requiring a lot of maintenance, especially if the resulting synthetic culture is intended for extended use over many, many generations. And previous research for controlling these phenotypic ratios in co-cultures have primarily sought to address this problem by manipulating and pairing two pre-existing biochemical systems. In synthetic biology, the first we use is quorum sensing, and this programs cells to secrete certain molecules, which we know as autoinducers, to act as a signaling medium. So once we've accumulated a number of cells and they've secreted a certain concentration of these autoinducers, this triggers a collective change in gene expression. With this, one example of how we can apply the system and pair it is by using this quorum sensing to activate transcription factors in the genes in order to activate cell lysis circuits that induce cell death, causing them to self-regulate and control, and this kind of becomes a method for us to control the population density or diversity however we want it to, preventing the propagation of any unwanted mutations inside our cultures and our systems. But of course, these proposed mitigation strategies, it's far from perfect, and some notable limitations that we've kind of noticed are that first, there may be a limited reproducibility if we're trying to apply the system to cultures that are using maybe less dense cultures. And in these kind of scenarios, because there are less cells, we may not be able to reach that critical threshold concentration, which I've mentioned. Secondly, programming these cells to actually facilitate the quorum sensing and all that, we're adding on like metabolic burden that may be avoided if we try to use alternative solutions. And finally, with all these methods, yes, it seems like there's a way out, but then we're also aware that this is still not protecting the cells and the cultures and the genes from microevolutionary changes that could affect the responsiveness and the efficiency of our circuits. So this is where we come in. We're asking, are there other solutions out there that we can try? What if, instead of aiming to really have an absolute order and control over our synthetic organisms, what if we just embrace the idea that there might be things that we don't need to control? And what if we start leveraging on this chaotic unpredictability that cellular processes inherently are like? We're suggesting this because if you think about it, intentional chaos and changes on a genetic level are actually not unheard of. In fact, life does have intentional systems in place that are designed to just shake things up, shake up the whole the whole bacterial genome. And you'll see what I mean once I pass the time to my teammate Eric, who will tell you guys more about our project inspiration and design.
Thank you, Stephanie, for starting with the introduction. So now I would like to shift your attention into how we got to develop our topic, which is novel compared to the other past experiments. So our team was inspired by Brainbow, a groundbreaking development in the field of neuroscience back in 2007 by scientists at Harvard University. So they designed a genetic construct containing instructions to produce several fluorescent proteins, such as red, green, or blue. So these genes were paired with recombination sites, which upon stimulation could cause random rearrangement within the construct so that they only one of that many fluorescent protein would be expressed. So the result was very interesting. The individual neurons expressed different fluorescent color signals, despite being provided with the exact same genetic circuit. So this allowed highly efficient tagging of each neuron for visualization and analysis in the future research projects. So this here, we saw it as a glimpse into what was possible. So we would like to have a heterogeneous population of cells expressing mutually exclusive characteristics, but without the inconvenience of maintaining a complex cell co-culture. So rather than trying to individually manipulate each cell, we thought of trying to first give our cells the instructions to express all our desired phenotypes. Let's like, for instance, like A phenotype or B or C. And then we use the modified Brainbow construct design to randomly induce the expression of certain genes. So we started thinking, how could we potentially adapt this Brainbow principle to our bacterial culture? So one key issue we need to address was that how the initial function of the genetic circuit could be conserved, like maintained throughout the generations, bearing in mind that nerve cells and single-celled organisms like bacteria and yeast work differently. So we needed to figure out how this principle can be conserved throughout the generation, and I'll explain how we came across with another unique inversion trait of Krylov-Speed system in this following slide. Okay, so to clarify, our project aims to ratiometrically control in a mixed culture, which is like a differentiation effect in eukaryotic organisms, say for an example. So the basic idea behind this concept is that instead of using bacteria in a monoclonal population carrying out different tasks by itself, our system works like a society where there is a division of labor to increase the productivity by reducing the individual's metabolic burden while increasing the product yield. So to specifically tackle this problem, the design we utilized in the original gene circuit of Krylov-Speed recombinase used in the Brainbow experiment, sorry about that, needs to be modified for our desired goal. So it does not include the excision design. So, I mean, in the previous slide, the Brainbow used the excision event to randomize the phenotype. But this time, instead of using the excision event, we'll try to make it capable of being reversible by maintenance of genes in circuit. Okay, so before I move on to explain the details of Krylov-Speed regulation, I'll start the general rules that we need to keep in mind. Okay, so the general rules for recombinase-based circuit is that first, Cre is not toxic. As you can see on the right photo, cute illustration, Cre, even though it's overexpressed, we want it to be Cre mutant, so it can be efficiently bind to the lock sites in bacteria. So another important rule is the orthogonality of sites. So which is on the left side of the presentation. So we need it to be 100% guaranteed with the independence of different sites so that recombination effect only happens within that specific site pair. Okay. So additionally, the reason why we are using Krylov-Speed system is that it's advantageous for over many reasons. So Krylloxb recombinase's advantage in gene regulation would be that, like, first, that it could lower the metabolic burden of individual bacteria by having it express only one phenotype at a given time. So, and secondly, the genetic circuit could function like a backup plan, like a backup memory, which it's capable of switching on its own gene once it needs to be. So it's more likely to be resilient to the specific gene mutation on specific phenotype, since it can replace it and have other phenotype to back up. So thirdly, Krylloxb system would not be affected as much for the extracellular environment and its own cell density. So this system could be suitable for many applications due to this versatility. So now I would like to discuss like more in depth of Krylloxb based regulation. So there will be two plasmids, each containing Kry enzyme and reporter gene surrounded by the lock sites. Okay, so once Kry binds to each lock sites, they will inverse the flip, the lock sites, which the inside there's a gene will be inversed. And this will happen for over many different generations, which means because of the inversion event itself is randomized, as time passes, the outcome ratio will converge to 1-to-1, 50% each. So here is a two graphic representation that demonstrate what I'm trying to explain. So I compare one with normal mixed culture of two different metabolic burden metabolites, and next one follows with the narrow, the Krylloxb regulated system. As you can see from the right graphic representation, it shows the divergence of two species due to their metabolic differences. After some time, you can see that metabolically less burdened species would over-dominate the population. Okay, and this here is a general representation of how natural selection works in nature, right? And this phenomenon was what we're trying to solve using recombinase system. So for referring to the left graph, as you can see, equilibrium position can be achieved using our system. So even if the population originally favors the less metabolic burden. So moving on to the next part. Now I'm going to discuss more of the dry lab aspects, which mainly involves programming. So firstly, we need to determine the constant values for the input data. So in order to test out systems efficiency, we'll use different fluorescent reporters that would be able to let us label and characterize different cells. The first experiment would be done with very simple systems that allow us to understand how efficient our system is, which is the rate of Kry enzymes, which dictates the rate and place of equilibrium position. After the collection of that data, we would have to build more specific or complex circuits with more fluorescent protein that would achieve different ratios. Computer simulation and sequencing library would be built on all the possible arrangement and also phenotypic ratio that could be achieved. Okay, and this will be utilized by researchers. So here, we're not only limiting the genes to be flipped, but also other gene elements like promoter terminators and could also be incorporated in the system for diversification of designs and hence the outcomes. This can function as a custom-based library, which researchers gain their best suitable circuit for their specialized experiments. So to make it suitable for researchers, we would first have to collect their own research data, and from that, we would build and test out the system's stability to determine all the possible parameters for the model. For example, we would test model without our construct first, and after determining the natural metabolic burden that happening in that experiment, and after deriving that constant, we will also derive other parameters that will be affecting our system from the experiment, and we would then introduce our recombinase system and see whether our system could save a declining cell population and loss of phenotype. So that is it for our project development. And now my fellow friend, John, will explain the plans for future iGEM plans. Yeah.
Right. Thank you, Eric. And now I would like to talk about the progress and the future of our project. So we are currently thinking of extending our project to a two-phase project. The two-phase project is process of extending our current project for a year to gather more detailed and sophisticated data and results. At this moment, due to the restricted access to labs caused by the COVID-19 crisis, our team has decided to put more focus on the dry lab for this year. However, we will still try our best to construct and clone through circuits, design, containing the Cree-Loxby recombinase system. When the pandemic situation resolves next year, then our team plans to carry out majority of the project through the actual wet lab, doing various physical experiments. So our current planned goal after confirming the success of the mentioned recombinase system will be applying our system into a real life example. We are currently considering about cooperating with other Hong Kong iGEM teams by applying our synthetic circuit design of recombinase system in their projects. We have gone through some of these ideas already with other teams. For instance, just to briefly introduce, Hong Kong City University team is currently working on ways to produce different types of plastic degrading enzymes from engineered organisms. During the process of producing different kinds of enzymes at once, it might be important to produce right ratio and amount of product together at the same time. Since some enzymes will have high metabolic burden, while some will have relatively low metabolic burden, over time, the cells with low metabolic burden will outnumber the cells with high metabolic burden, which will therefore lead to uneven production of wanted enzymes. Therefore, by adopting our recombinase system design and putting it into a cell, we could potentially bring stable ratiometric control and produce wanted ratio and amount of enzymes over a long period of time without experiencing a loss of one phenotype due to the cells with high metabolic burden being over-dominated by the ones with low metabolic burden. Furthermore, the current synthetic circuit design of recombinase system could also potentially be utilized into the topic carried out by Hong Kong University of Science and Technology on producing fabric through putting two different protein chains together. One of the key factors in this project is to produce two separate protein chains, and different polypeptide chains will have different metabolic burden, which could potentially lead to uneven production of desired proteins. Our team believes that the complex design of recombinase system with the protein coding gene could potentially lead to consistently producing the desired product without experiencing the loss of one protein chain producing cell due to different metabolic burden. So plan beyond the real life application. So we are currently working with DH5 alpha, which is one strain of E. coli. We are hoping to implement this synthetic circuit of recombinase system on the eukaryotic yeast, which is known to be more complex. We are hoping to receive some help from Dr. Karen Yu, the specialist in yeast biology in HKU. Moreover, based on the data collected by utilizing such system on yeast, our team wishes to further apply it in more complex mammalian system so that it could potentially be used for integrated human practices. Other than just changing the type of organism, our team is also planning to operate this synthetic circuit with other recombinase systems besides Cree-Loxby, such as lambda red and FRP-FRT system. So to conclude, the main purpose of our synthetic circuit using the Cree-Loxby recombinase system is to stabilize the different metabolic burden of individual cells and to achieve the desired phenotypic ratio. Our team has currently been working on the prokaryotic cells, but plans to extend the research to the eukaryotic cells so that the system could be used in more practical aspects such as biotech industry. The potential real-life application that is being planned is implementation of this system to the production of various types of plastic degradation enzyme being worked by City University team, and the production of two different types of protein chain to create protein-based fabric, which is being worked by HKUST team. Beyond the degree of simply stabilizing the metabolic burden, in the future, our team also wishes to achieve more complex things such as auto-editing, such as gene knock-in and out in the synthetic circuit by the cell itself through utilizing complex design of various types of recombinase systems. This will, of course, need a huge amount of computer work and dry lab. So before we finish, we would like to mention about the various collaborative works and human practices that our team has been working on. Besides this symposium, we have organized various online and offline meetups with other iGEM teams in Hong Kong and have talked, troubleshooted our projects with ideas, project and ideas for further collaborations. We hope to further cooperate with various experts and authors of critical research papers for improvement of our project. We hope that our current topic could potentially bring benefit to the field of synthetic biology and science. We would also like to thank Dr. Jay Zhang, various faculties and professors of HKU for technically and financially supporting our team so that we could host this symposium. And thank you for listening.
Okay, thank you, Stephanie, Eric, and John for presenting for us. So we'll now open to the panel. So any panel members, please give us some comments and ask some questions.
I just have a very simple question. So apparently this project is trying to tackle the issue about metabolic burden for bacteria, so that you can use bacteria to consistently produce whatever you want. So is there any safety concern about this? Will you try to, is there a chance that you're trying to accidentally create a superbug or something?
Well, I think first of all, the phenotypes that we will design and to implement in our circuit would not really cause any like environmental harm. So it's very essential that we have to test the Cree enzyme out first with the recorder gene first, and then we'll try to apply that with other genes. But then we wouldn't want to like use some like harmful gene so that it can be exposed to the environment. So yeah, but then I'm pretty sure the mechanism itself wouldn't, unless it creates like mutation, I'm pretty sure it's okay.
But to build on that response, I do understand. I think, thank you so much for that question. And it's a very intriguing, I guess, way of thinking about it. And the way we can possibly install different genes, perhaps like antibiotic like resistance and different kind of switching around. I think it's also viable prospects. Like we have to really acknowledge that that is possible, and but it's also a new avenue of like maybe how we can also extend into like medical research and use this system there. But yeah, I think you've given us something to think about, and it was really, really interesting.
Yeah, I guess that's one of the point that not many teams is trying to mention is because if I remember correctly, one of the element in iGEM is that you have to consider about safety.
Indeed, the safety and ethics the concept. I think yeah, that's
yeah. Thank you.
Okay, I appreciate it.
And there's a question from Felicity. Or would Felicity want to unmute yourself?
Oh yes, I would like to actually repeat the question first. So the question is that if it's possible to control the probability of each gene to be expressed. Yeah, so after testing, currently the experiment that we're on is to express 50-50, which is the 1-to-1 ratio. But then we could actually design a more complex genetic circuit, and we'll try to do that using a computer, like a programming simulation with different log sites, and maybe we can use different like a FRT, FLP FRT, and then Relox, and we can have a combinations of it, and then we can derive the different ratio based on the modeling. So yes, it is theoretically possible.
So do you mean that different Relox sites have different chance of flipping genes, and then you can derive the exact ratio through modeling? Is that correct?
So, I mean, the efficiency of recombinase may differ. So I think we have to first set the parameters for our modeling equation, and then we can adjust it based on the programming. So yeah.
Okay, understood. Thank you very much.
Do we have other comments or questions? Okay. There's one more question. So Richard asks, will you be able to ensure a one-to-one ratio if you use the same promoter type to drive two different genes?
Okay, so I think it's not the matter of promoter, but we already set the promoter. I mean, promoter can be constitutive or it can be inducible, but the genetic construct itself is it does not really for our first experiment, which leads to one-to-one ratio of two different genes. We want the promoter to be outside the lock sites so that it does not really affect the outcome ratio, but the ratio itself is only determined by the overexpression of Cre, and by Cre binding to the orthogonal lock sites, it can perform a flipping event. So, like the flipping itself is randomized. So, over time, as the population grow, it will converge to one-to-one ratio. So that's the whole point of what we're trying to achieve.
So we'll wait for 10 more seconds to see if we have other questions. Okay, we've got another question. If it is a random event, how would you believe that it would eventually go to one-to-one?
Okay, so to explain, like it's kind of like a probability question. So let's say you like flip the coin, right? Let's say one side is head side and another side is tail side. I mean, you do it for over very many generations. Like you're flipping the coins for like 10,000 times, and eventually the ratio will be one-to-one, right? The heads will be like, I don't know, 50% of that, like the chance of that outcome. So the general idea is that because bacteria has this rapid growth and replication, so it can, you know, eventually converge to that one-to-one ratio. So.
Yeah, okay. So if there are no other questions, thank you for the presentation. And then now we will move on to the presentation from the CityU team. So CityU team is currently working on a project, biodegradation of plastics, a rational design approach. So the importance of plastic and the dire need to find a solution to combat plastic pollution are two opposite sides of the same spectrum that one can't deny. So through the project, CityU proposes to design and develop a technology to facilitate biodegradation of plastic waste through integration of genes of plastic degrading enzymes such as papain and specific types of peptase, lipases, and esterases, and make it a multi-plastic eating bug by a site-directed mutagenesis approach. So on behalf of the Hong Kong CityU team, Delta, Ellis, David, and Meng will now present their project. CityU, please.
All right. Good afternoon, everyone. This is Hong Kong CityU iGEM Team 2020, or the Team Pluslicious. Small team with 9 members, all from the Department of Chemistry. Let's get started. Our presentation consists of 2 parts. In part 1, we will mainly talk about the plastic problem nowadays, including the plastic problem in the living environment, the challenges from them, and combating with the issues. On part 2, we'll more talk about what our project is, including computer-based experiment and mutagenesis, and the other advancements. Global plastic production is increasing. The statistic from 1950 to 2015 shows a drastic increase of the plastic production. For example, in 1950, 2 million tons per year of plastic was produced. And 3 years ago, 2015, 381 million tons per year of plastic were produced, about 200-fold of increase. Well, it's reasonable. Plastic is necessary these days. Many things that we use in our daily lives, including plastic bags, bottles, disposable lids, and straws, they're all made of plastics. Thus, we have high demand of plastics. Let's look at this graph. As you can see, all types of plastic waste, including PET, PUR, polyurethane, this one, has been increasing with the increasing year, which is due to the high demand of plastic. 6 billion metric tons of plastic were disposed since 1950s. The consequences are creation of the odor, generation of the leachate, and production of landfill gases. Now, what if this tremendous amount of plastic is disposed into the ocean? Well, there will be also the leakage of solid waste from the plastic waste, but also marine animals will fall victim. For example, look at this turtle. This turtle's digestive tract was filled with plastic. Therefore, it was unable to consume the food for survival and starved to death. These are the problems that we can see in the environment due to the plastics and gaps in the current solutions. Regarding this part, Ming will explain more.
So when it comes to plastic pollution, we should never underestimate its seriousness since no one can get off the hook, and current technology can help alleviate the issues, but improvement may be needed. And that's what our experiment plans to fill the gaps in current solution. So let's go into details. So this flowchart indicates the phase of the plastic waste, and from there you can see that a majority of waste will either be discarded or to be incinerated, and only a few of them will be undergoing the recycling process. Oh, and from this slide you can see that the public awareness on recycling has not been raised so much. In 2015, only a mere 3.5% of the recycling rate is increased, and then compare with the data in the previous 5 years. So here is one of the examples showing that the process by which plastic bottles are recycled. The first step will be label detaching to remove the label before it can be crushed into small pieces. So in the third step will be material separation to separate the caps and bottle materials because they are made from different plastic. And after that, the plastic flakes are dry and then extruded to make long tubular strands. And the strand will finally be cut into pellets as raw materials for new products. So the new products can be Pilo and Crow. So this slide, I will talk about the existing problem on handling the PET plastic. So the first one will be as you can see before, the recycling has not been widely practiced and the recycling awareness is not as strong as we expect. And the main approach to deal with the plastic will be disposals. And this is because people always have to return the plastic products not in use to the collection point, and this is very inconvenient. And also from the previous slide, you should also notice that over 15% of plastic waste are discarded to the environment, and this can lead to long-term impacts, say for example, the landfill saturation and ecological crisis. And these are the existing problems on handling PET plastic. Until now, the ongoing treatment on PUR plastic is tertiary recycling and to make it become raw materials, and then they can be further chemically synthesized into new products. But it can produce some toxic byproducts, for example, aldehydes, ammonia, cyanides, and nitrogen oxides. And also the environmental pollution may exacerbate and threatens humans' physical health. And therefore, biodegradation of plastic seems to be an ideal way to turn PUR into less harmful substance. And that's why we will focus on these two plastics, PET and PUR. So you may now wonder what has inspired us to focus our lab work on plastic degradation, and there are two main reasons. So the first one is to investigate alternate ways to tackle increasing plastic pollution using engineered enzymes. And the second reason is that there is scarcity of research on the breakdown on PUR and PET plastic. So now I will leave the experimental part to Delta, please. Okay, so let's move on to our experimental parts. So in this year's projects, we adopted a two-step approach. First, we will modify some enzymes, and then in the second step, we'll analyze those results with some docking analysis. In our experiment, we plan to genetically engineer the bacteria to produce some modified plastic degrading enzymes. We create them through in silico mutagenesis. And at the end, we will create some new biobricks for future studies. And all the results are evaluated by some docking analysts using AutoDocWinner or Pyrex. And the interactions between enzymes and substrate are also examined. So this is our experimental flow. And we first identify the amino acid sequence of our target enzymes first. And then we either get its 3D structure from LCSB PDB or using a modeling software called Modeler. After that, we will perform our first docking to obtain an affinity of the wild type enzyme. And probably we will identify a possible binding site. And we will do a virtual mutagenesis afterwards, and we will do the docking again. If the affinity after the mutagenesis is increased, and then we can use that mutant to make a new biobrick. So we have 5 targets of enzymes. We have 5 enzyme targets. They are first polyethylene terephthalate hydrolase, also known as PTase. And the second one is papain from papaya. And the third one is polyurethane esterase from Coromonas. And the last 2 are also from Pseudomonas bacteria. They are polyurethanase esterase A and polyurethanase lipase A. So among the 5 enzymes, papain can react with 2 types of plastic. They are polyethylene and polyurethane. However, the affinity between papain and polyethylene is not really high enough. So we'll focus on the papain and PUR binding instead.
and we choose the texturemic form of plastic as our substrate. On one hand, it is long enough to do the simulation, and on the other hand, the time spent for docking a tetramer is much more shorter than docking maybe a pentamer or hexamer of a plastic. So we can find the PDB file of peptase and papain from RCSB PDB. So we directly use the structure as the 3D structure of the enzyme. However, for the rest of the enzymes, we cannot find the structural file from the database, so we need to do a homology modeling to find out the 3D structure. First, we need to input the query sequence of our target enzymes, and then we need to find out a template structure that has a similar sequence compared with our query sequence. And then we input these two files into the homology modeling program called Modeler, and the program will automatically generate a homology model. And then we can use this model to perform our dogging or our in silico mutagenesis. After finishing the first docking, we will continue to do the in silico mutagenesis. According to some research articles, the highly conserved region on either a loop or a turn on a protein is quite important for the binding of substrate. And we perform a multiple sequence alignment to locate those regions for us to perform the mutagenesis. And after that, we will also use another program called legplot. So what's legplot? Legplot is a program that can convert a 3D output file to a 2D plot. And then all the interactions between the ligand and the enzyme can be visualized. And after we perform a multiple sequence alignment and visualizing the interactions using legplot, then we can make some mutants. And we have made 2 mutants, and we will discuss this later by our and other teammates.
Thank you, Delta, for the explanation of the computational studies. We changed the glycine to tryptophan in papain, and we changed arginine to phenylalanine in PUDA. We hypothesized that if a hydrophobic residue replaces a polar residue, the binding cavity's hydrophobicity can be increased. Then the intermolecular forces between the non-polar substrates and an enzyme can be greatly enhanced. Finally, our experimental results agreed with our prediction. After the in silico mutagenesis, the predicted affinity calculated in delta G were improved from -7.6 kilocalories per mole to -8.6 kilocalories per mole in papain and -8.2 kilocalories per mole to -8.8 kilocalories per mole in PUEA. This indicated that the substrate binding would be more favorable after the in silico mutagenesis process. Finally, our experimental to further improve the efficiency of the polyurethane plastic belt degradation, we can implement a combining strategy. For this reason, we can insert several forms of polyurethanase genes into a plasmid vector, including papain, PUEA, PUDA, and PULA, to express multiple enzymes from the E. coli. Also, we use a new promoter to increase the transcriptional activity of our mutant enzymes. This enables our bioengineered E. coli to express multiple polyurethanase to deconstruct the polyurethane more efficiently. In conclusion, the plastic pollution crisis has been a persistent environmental issue, and we cannot cope with the massive increase in the number of plastic waste on our planet. One critical step to solve the problem is to use our knowledge in silico mutagenesis and use the knowledge to manipulate the gene expression. we have known about the natural enzymes which are capable of plastic biodegradation. And our team has produced the molecular docking data between those enzymes and different plastic polymers. After that, we use computational enzyme design to generate more effective mutant polyurethanes that were strongly supported by our in silico docking studies. There was an improvement in the affinity of our mutant enzymes. Our new gene constructs may also probably shorten the duration of biodegradation of polyurethane plastic. Ideally, our recombinant E. coli would provide a new recycling hope of common plastics. And these are our reference. And if you have any further questions, you may try to contact us via Instagram, Facebook, or email. And last but not least, we conduct a survey on synthetic biology. If you're interested, please scan the QR code below and please feel free to complete the survey and let us know your thoughts. Thank you very much.
Thank you, Delta, Alice, David, and Ming for your presentation. We would now like to request the panel members to provide any feedbacks or ask any questions if they have it. Okay, so we have one question from Mark Rousseau. What is the biggest limitation of in silico mutagenesis in your opinion?
I guess one of our limitations is that we still need to perform the wet lab studies to prove our data. Is because it only shows the predicted affinities of the docking between the enzyme and the plastic substrates. And on the other hand, however, since the iGEM, due to the limitation of the COVID-19 outbreak, we can only do as good as possible to do some more accurate in silico docking by putting those results into several docking program or servers to predict the most accurate model of the docking.
So then we have another question from Judy. How much is the increase in affinity affect the enzymatic rate compared to the original rate?
I guess the answer is like the same. Due to the limitation of the COVID-19 outbreak, that we still need some wet lab process in order to identify the rates of the enzymatic reaction. However, the affinity, there are some literature research shows that those enzymes actually had an ability on the degradation of those polyurethane plastics.
Okay. So then we have a question from Dr. Simon Xu. If you can get back to the lab of the experiment, what will be your plan?
Actually, our plan will be we will do the mutagenesis in our wet lab, but however, there are still some limitations because we still need to figure out how we can measure the efficiency of the plastic degrading. And just like the question asked by Judy, we still need to investigate how the increase of the affinity can affect the enzymatic rate. And after we find out that how to analyze the results, that is, we can find a relationship between the the affinity and the reaction rates, then we can really work on doing a real mutations into the onto the protein, and hopefully we'll get a positive result. So this is our plan in our stage 2.
So then we have another question from Mark Rousseau. How will you envision the technology being applied to solve the plastic problem?
Actually, we haven't thought of this problem, these questions of how we can apply our technology into solving some the plastic problem. But I think that the direction should be after developing the plastic degrading bacteria, and we can hopefully maybe to design a device to maybe like a recycling device that can use our bacteria to degrade the plastic waste into some other simple product that can to use as another purpose.
So we've now got the question from Kenneth. So Kenneth, can you unmute yourself?
Hi, I just want to ask, because like proteomics usually is very hard research. Usually it requires a lot of data and also machine learning from like experimental data, like X-ray crystallography, and also mass spec with a kind of cross-linking to determine the 3D structure. So I kind of have some suspicion about using in silico to do a mutagenesis library. And also I have a kind of a question. I'm not very familiar with this field, so I want to ask, like, is affinity directly proportional to the activity of the hydrolase of the peptides? So is there like any paper proving that? So so it's like you're trying to increase the affinity, but does it really increase the activity? I hope there could be some paper supporting that. And also, actually, there's a lot of mutants library available outside. Would you like maybe consider also trying those in your construct to compare? Yeah, and also maybe do docking with those kind of construct to see if there's really like a direct proportionality to compare using the model.
Okay, so for the first questions that you asked, that is a modeling 3D structure is reliable or not? We can say that it is not as reliable as the X-ray, the structure you obtain from the X-ray crystallography or mass spectrometry. However, we have also done some process to make the model more reliable. We have do some edge minimization or some other process to actually realistic the model. So using a predicted 3D structure is not the best way, but is the most suitable way to do in our stage, since we can't really do a crystallography to know the real structure. And for the second questions, I think if you're asking that is the binding affinity is directly proportional to the activity, I think that the affinity should be directly proportional to the activity periodically. However, I think that we still need to have some wet lab data to support our support this because we only doing the dry lab is not really sufficient to prove our hypothesis. We can say that the affinity increased, that the activity will also increase.
Okay, we have a question from Paul San. Why do you need the tetramer of PET in your docking experiment? Would the final variant of your enzymes be specific to the tetramer but not others?
We have explained it in our presentation that because the tetramer is we think that it is quite long and is long enough to do the simulations, and on the other hand, the docking time is also crucial because if we use maybe octamer or even longer polymer, the docking time will be too long, and it's not really realistic to do a long polymer chain. So we choose that tetramer as our substrate, and there is another reason that we choose tetramer is because we have found another research article that is studying on the activity of the PETase, PETase, and they are using the tetramer. So we think that tetramer will be a suitable candidate for docking analysis. And the second question is.
I think the last question is from Hannah. So maybe if we can request Hannah to unmute, and then maybe you can answer yourself.
Yeah, I think my first question is similar to that asked by Dr. Chiu. I think we just wanted a more solid plan for wet lab. So in a two-phase project, maybe you will have to discuss your limitation for now and then present a more solid plan for the second year. So so in details, what are the tests and experiments that you determine the affinity and the reaction rates after you do the mutagenesis? And are they going to be measuring the weight or observing the pieces of plastic under the EM or something like that? Yeah, so actually, this is maybe a comment because there are a lot of iGEM teams that have worked on mutating and expressing PTAs or other plastic degrading proteins that are also expressed in mostly E. coli. So how is your project different from theirs? Are there any specific application that your gene, your plasmid constructs will be used, or would you consider improving other teams' project instead of like building a new one that's not very special?
Actually, in the first questions that you asked, that how can we test the affinity and reactions? Actually, we have some ideas. We'll probably test the efficiency of the enzyme maybe by measuring the weight, and also we'll also use the electron microscope maybe to investigate the structure before and after we put the enzyme on that polymers. And for the second questions you asked, that what's different from others is because I don't think there is any past iGEM teams are working on our enzymes such as papain or polyurethanase, other enzymes. They work on some enzyme like pectase or like the high school team, they work on cutinase. Those are some common enzymes. However, the papain and PUEA or PULA is not a common one. So I think that this is the major difference. And moreover, we also plan to construct a biobricks that consists of multiple segments of the genes in one biobricks, and then so that the bacteria can express all our proteins at once. So I think these two are the major differences from other past iGEM projects that are working on plastic degrading protein or something like that.
Okay, so thank you for the presentation and also for the questions. Because of time constraints, we'll move on to the next presentation. And if you guys still have any more questions, then we'll have a short discussion session after the last presentation, so you guys can discuss them. Okay.
Yeah, and thank you, Paul, for giving us the question in the chat box. We can address that maybe
in the discussion session.
Yeah.
Okay. So we will now have the presentation from the HTUST team, who are working on the project Green Textile System, Hardfish Intermediate Filament Production and E. coli Exploration of Chromoprotein-Based Dyeing Method. One of the biggest and most harmful textile pollutants is microfiber, a synthetic fiber with diameter less than 10 micrometers, which mainly comes from washing and disposing of clothes. Through their project, HTUST proposes to synthesize hardfish intermediate filament, a biodegradable and high-strength green alternative to existing textile material. They hope to redesign fashion in an environmentally friendly way by expressing the intermediate filament gene in E. coli to synthesize it. Their project aims to work out the most efficient means of production for hardfish IF, as well as implement a color mixing model to dye the thread. On behalf of the HTUST team, Timmy and Derek will now be presenting their project.
Good afternoon, everyone. Thank you for staying too late. I'm Timmy, a member of the HKUST iGEM team members. Hagbrick. What's Hagbrick mean? Read after me. Hagfish, 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 hagbrick. Once more time. Fabric, 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 hagbrick. Yeah, now you know how hagbricks come from. It comes from two individual words, hagfish and fabric. Today, we are going to introduce to you a green textile system, hagfish intermediate filaments production in E. coli, and the exploration of chromoprotein-based dyeing methods. Sorry, wait a minute. Okay. Our project aims to ease two major environmental issues. The first one, microplastics. The second one, dyeing pollution, by producing social applicable protein-based fibers with good physical properties and vivid colors. Let's go to the first environmental issues we want to solve first. Microplastic problems. Yeah, plastic again. I believe everyone should have heard of this, but let's think about it. Where do this kind of tiny plastic came from? On your textbook, you may know that it may came from degraded plastic bottles, plastic bags, or even microbeads from your facial wash. But you might not be aware that actually 35% of primary microplastics in the ocean came from synthetic clothing. These kind of microplastic are called microfibers. These fibers wash off from your clothes, enter the ocean directly without being treated by your washing machine or the municipal sewage treatment scheme. These tiny pin-like pieces could even poke cells. More than 700,000 pieces of microfibers are released to the ocean every load of laundry on average. Now, let's check your clothes and wearings to see if your clothes are made up of the following kinds of synthetic fibers that could cause microfibers problem. The first one, spandex. It is a kind of elastic materials usually used in the production of leggings or performance wear like swimsuits. The next one, more common, nylon. It is usually used in the production of t-shirts or waterproof jackets. Top of the rank, acrylic, or is called polyacrylonitrile PAN. It is a cheaper substitute of cashmere. So it is a substitute of wool. Common products used PAN to produce is scarf and wool jacket. What's the fate of microfibers? Identical to every kinds of microplastics, once it enters the ocean ecosystem, it bioaccumulates and magnifies along the food chain. And the end consumer, which is human, suffers. Look at the map behind. Thanks to the extremely small size and lightweight of microfibers, microfibers could be transported all over the ocean by ocean currents and cycle through the water cycle. It is literally everywhere. So you can find microfibers in your table salt, in your alcohol, and in the water you drink every day. Such an important environmental issue must have several solutions. There are two kinds of filtration system aimed to solve this kind of microfibers problem. The first one is membrane bioreactor. It is a filtration system that could be adopted in a city's sewage treatment scheme. It is very effective that could eliminate up to 83% of microfibers. But unfortunately, Hong Kong is still on the very top figures, is using the secondary wastewater treatment system, only dealing with 44% of the fibers. The second one is a filtration system that could be directly fit into your washing machine. These two kind of filtration systems are both very effective, but rarely can you see people using this kind of system. Why? The point is money, because this kind of system is very expensive to install, and the maintenance cost is also very expensive. It comes to a little bit brighter side of solution. Protein fiber substitution. The fibers shown in the figures is spider dragline silk. It has unparalleled elasticity and tensile strength, and because it is made up of protein, it is completely biodegradable. It looks very perfect on paper, but why didn't we saw a piece of clothes that is made up of spider dragline silk on the market now? It is because of its nature of the protein. Protein making up spider dragline silk are very huge, and it is very highly repetitive in nature. It makes that the expression factors feel very difficult to catch up with the expression and the production of proteins. Here, Hagfish kicks in. Introducing Hagfish slime intermediate filaments. This kind of intermediate filaments is 13 times more extensive than Kevlar, 4 times more stiffer than nylon, but only 1/4 the size of spider dragline silk proteins. So in theory, it could be produced in expression factors more easily. Talking about hagfish, what is hagfish? This is hagfish. It is a scavenger living in the Banff Lake community. When it's irritated, it secretes slime. It is used to clog the gills of the predators to escape from predations. Let's look into the slime. There are two components. The red one is slime threads, and the purple one is mucin strands. In our project, we focus on the red one, slime threads. We also call it hagfish slime intermediate filaments. It is a dimeric keratin-based threads made up of two subunits, namely the alpha subunits and the gamma subunits. In our project, we are using Hagfish IF from the Pacific Hagfish. So the pink block is showing the two subunits. It is worth noting that because it is made up of two subunits, we will have two plasmid rather than one. And for the purification process of the proteins, we are using his-tag purification. And also we intentionally add a TEV protease site between the subunits and the his-tag, because eventually we would like to eliminate the his-tag from the subunits to prevent it from jeopardizing the foldings of the IF. Because our project is in the manufacturing track, we would also like to boost the yield of the proteins. We will boost the yield by trying different promoter and RBS set. And also, we also incorporate a TOR-A signal peptide into the sequence. This signal peptide could export the protein to the periplasm to avoid the formation of inclusion body. Our primary chassis is E. coli, but we also do codon optimization for a yeast because of after consulting a hagfish doctor, a doctor working with hagfish slime, we found that it could be difficult for a prokaryotic E. coli to produce a eukaryotic proteins. It comes to the second environmental problem we would like to solve, dyeing pollution. Apparel dyeing is the second largest causes of water pollution. So every clothes you wear after dyeing is discharging nasty water, but you won't want to wear a clothes without color, right? So our project are also working on the dyeing perspective. The first dyeing method we are going to use is directly linking chromoprotein. Chromoprotein is CP. Directly linking chromoprotein at the N terminal of each of the subunits through peptide bond. The second one is directly mixing chromoprotein with our non-color IF. And the third one, we will try out organic dye to see if the color is vivid enough and if it will jeopardize the physical properties of our slime threads. Seeing this photo, you can see that we reserved a restriction site ACC65I for chromoproteins. Sorry. Because we have two subunits, each subunit can bear chromoprotein of different colors. So this is what we call color mixing. If the alpha subunit is linked to a red chromoprotein and the gamma subunit is linked to the blue chromoprotein, we hope to see a violet IF formed. Following is our experimental rundown. Our experiments would be divided into 3 stages. First, the expression and purification. After culturing the cells in a LB medium, we will perform his-tag purification and check the protein concentration. We will also cleave out the his-tag residue after the purification, and as the reason I've mentioned. And the second step of our experiment would be thread formation. We will first perform a stepwise urea dialysis for the IF assembly, and then followed by the preparation of protein dope solution. And after that, the fibers are ready to fabricate. After the fibers are fabricated, we still need another step to enhance the mechanical strength of the slime threads by draw processing or glutaldehyde cross-linking. Here comes the dry lab part of our project. Derek, please.
Thanks, Jimmy. So this is Derek from UST iGEM team. So I'm going to talk about the molecular modeling in our dry lab part. Next page, please. So first, I want to reintroduce the tensile properties of our hagfish slime intermediate filament. So The native hagfish slime threads exhibit intriguing mechanical performance. And a notable feature of these threads is that their mechanical response can be tailored by a jaw processing mechanism mentioned before. So as you can see on the right graph, the optimally processed threads exhibit enhanced mechanical properties, which is comparable to that of spider silks and even approach high-performance composite materials like Kevlar. So you may wonder what is the molecular basis of such kind of enhanced properties. The answer is alpha to beta transitions, which is essentially a universal deformation mechanism in alpha helix rich protein materials such as wool, hair, and also in our IF filaments. So this transition in hagfish slime threads has been confirmed by wide angle X-ray scattering as shown on the right-hand side. However, is it possible for us to visualize this dynamic jaw processing from a micro point of view and understand the alpha to beta transition of the constituent proteins along with it? So we use molecular modeling to achieve our goals. So our work is divided into two parts. First is protein structure prediction, and the second is molecular dynamics. So here are three objectives of employing molecular modeling. First, we want to predict the molecular structure of hagfish slime IF subunits and dimer. And second is to estimate the tensile properties of our product. And third is examining the thermodynamical and conformational stability of fusion proteins mentioned before, and we are still working on it. So here is the flow chart of molecular modeling work, and I will talk about each step in detail in the following slides. Next page. So about the first part and first step is predict protein structures based on sequences. In the UniPro database, we found that there are only amino acid sequences of our subunits, but no 3D structure available. And from our interview with Dr. Douglas Fudge, who is an expert in hagfish slime, we learned that the terminal regions of IF are inherently disordered proteins, which may make it difficult to experimentally determine the atomic resolution structure. Therefore, we turn to the power of computation and algorithms to predict the 3D structure of our proteins. So we mainly use two protein structure prediction servers. One is Swiss model and the other is iTezers. Both are well-known servers. And we also adopt two approaches. One is to input two subunit sequences individually. And you can see yellow graph shows the alpha subunits model we get, and the red is the gamma subunit model. And the second approach is to input two subunit sequences together as a dimer. And on the right-hand side is the dimer model we get. So the next step is to analyze the quality of our predicting models. So first, we use inbuilt analyzed values like B factor, local accuracy to select those three models above from among the many candidates. And then we use external tools like ProSA and more probability to help us to analyze. So here we show the results from the more probability. So you can see that when it fails one criterion, the rectangle will turn green. So you may see that dimer outperformed these two individual subunits. So we set dimer as our prior models, but we still also have another direction is to combine two individual subunits models into one dimer model afterwards. Okay, next page. So here comes the molecular dynamics. So it is essentially a numerical method simulations in which the equations of motion are solved to study the physical movements of atoms and molecules in a system. So at each time step, the program will compute the forces acting on each atoms, whether it is bounding forces or non-bounding forces. and use a molecular mechanical force field. And in our project, we apply the charm force field developed by Harvard. And then we will update the molecular position, atom positions. Next page, please. So here comes the preparation work before the stretch test. So which are solvations, energy minimization, and equilibration in water. The reasons why these 3 steps are necessary is first, we want to approach the experimental conditions because whether in cellular environments or in vitro tubes, molecules are always right, our proteins are always surrounded by water, so we solvate it. And the second is to examine the stability of our predictive models. And third is because many protein structures come from X-ray crystallography at extremely low temperature. So the determined conformation may not be realistic at room temperature. So we'd better first do the minimization at room temperature. And on the right-hand side shows these 3 steps we use to do the preparation work. So first is to minimize the whole system for 6,000 steps. The second is to equilibrate the system in the NPT ensemble for 50,000 steps, which is exactly 100 picoseconds with protein fix. So the reason we fix the protein is to save the computational effort for the next step. So, and the last step is to free all atoms, minimize for another 2,000 steps, and run NTP dynamics for another 100 picoseconds again. Next page. So for the step 1, we can see from the total Gibbs free energy plot that it reached a relatively low level and keep a steady phase. And also value come from the algorithm gradient tolerance indicates that it also reached the convergence, or in other words, minimizations. Next. A second, step 2, we got a result also from the total free energy plot. We can see the energy goes up, but still a negative level, which means the system is still preferred, and it reached equilibrations afterwards. And on the right-hand side is the animation to show the simulation process. Next page. And step 3. So first, you can also see from the Gibbs free energy plot, minimize first and then reach equilibrations. And the RMSD is used to measure the accuracy of the protein structure. You can see these RMSD values reach about 1.7 plus or minus 0.2 angstroms, which means it's also reached a steady phase. Yeah, next page. And here comes the stair molecular dynamics, which is essentially to stretch the protein molecule in vacuum at a constant speed by applying an external force to simulate the tensile test in reality. So this program uses the equations of elasticity to get the force and the elastic potential energy we use in this model. Next page, please. So in order to plot the stress strain curve, we also need to get the force in the direction pulling, which we do that with the equations on the top. And we also need to get the strain at each time step, which we get with the equation at the bottom. And the scheme below shows just shows the process of this stretching process. We apply a force. There, the molecule get elongated, and there will be a delta L, and we, based on this delta L, we compute this string. Yeah, next page. So here comes the result of our. So at frame 1, which means at the beginnings, there are still alpha helical coidical structure, and when it comes to frame 12, random coils appears. And when it comes to frame 50 and 60, you can see those yellow arrows. These represent beta shapes. So we may demonstrate the alpha to beta transition from micro point away with this graph. And also we got the stress strain curve to show the tensile properties of our product, of our fiber. So our goal is to incorporate our result with other materials, with curves from obtained from other materials, and also incorporate the secondary structure of our protein at each frame to the our curve. Next page, please. So still something in progress is first is the IF bundle construction. We want to simulate the threads in the natures, and it's about the MD simulation at 70 Celsius degree because we want to test the heat resistance of our product as a textile materials. And also we will upload well community project files to the GitHub and hope to cooperate with other VMD users. Sorry, Timmy, you forgot to open your microphone.
Sorry. Thank you, Derek. Apart from lab, we also do science communication. We have created 2 comic series aimed teenagers and kids. For the former one, we explain common experimental mistakes and interest in an interesting manner. And the later one, we would like to raise children's awareness of marine and marine environmental problems by these cute cartoons. We also set up YouTube channels to upload several YouTube series like True Facts, Tammy's iGEM, and more. Stay tuned to our YouTube. Also, we have designed SimBio notes for Hong Kong students to help them to thrive in their DSE while learning more advanced SimBio stuff. While for collaboration and partnership, we have collaborated with the HK CityU team to hold various mini talks, and we are going to make different videos on related environmental problems. When it comes to CongU, it is our mission to adopt their Kree-lock system to produce our chromoprotein. It is worth noting that when we do codon optimization for the yeast, also considering to adopt their Kree-lock system because we know that Kree-lock system is not perfectly working in bacteria, but yeast is better. For our future goals, we would like to first have a light-sensitive circuit using wavelength as a stimuli to produce desired color of chromoproteins. And we also want an all-in-one purification system to replace the expensive heat-tech purification method. These 2 goals we are in active progress. Then we are going to find a better attachment mechanism of chromoproteins. And if we are successfully produce the slime threads, we would explore ways for mass production, extrapolating the production efficiency and condition to a manufacturing scales. You might ask why we seems to deviate so much. Because our team, rather than only producing a biodegradable and mechanically strong hagfish slime IF threads, we would also to consider the economical side of this textile system. We do not want a very expensive fibers that is not approachable to laymen. Here, and I'm going to end my presentation, and we are going to open for questions. Thank you.
Okay, thank you, USD team, for your presentation. So now we'll open to the panels for comments and the floor for any questions. So in the chat room, Kenneth posted a question. Maybe Kenneth, can you unmute and elaborate?
Yeah. So basically, the first I want to ask about the dry lab first. So you simulated the duplexed like a double protein chain, but however, like as I know, if you are going to make a textile, it's gonna be a network of proteins. So how is the interaction between different duplexed protein chains So because I think it's more on the network instead of individual molecules. So how is it going to interact with each other, and actually what's the mode of binding that they form a network of protein? And also the models constructed is with I see with a lot of water molecule, but then how about a dry version? Is there any way to simulate that? I'm not very sure about like how to do the modeling, but is it possible?
Hello, thanks for the question. And I'd like to answer your first question. So yes, so now, but you may also notice that in our in progress page, we mentioned like we want to reconstruct the RF bundle because we also know that the limitation of our preliminary results on dimer molecules is not enough, because we know that the RF exists in the nature as a bundle form or polymer form rather than a dimer or dimer molecule, single dimer molecules. So we are trying to construct the bundle form of our threads and also do the stress test on that. So because the dimer is also a unit for the bundle, right? So maybe also give some insights or just estimate the tensile properties. And about the second question, is about the dry version, right? So first, you may notice that the stress test is actually carried out in vacuum or in a dry environment. But for the solvation and energy minimizations, our goal, what we are thinking is try to approach the experimental conditions, because we know we incubate our fibers, or we want the fiber assembly happens in water aqueous environment. That's because they will simultaneously assemble into dimer or into polymer. So because we want to mimic that kind of environment, so we also want to do those energy minimization and equilibration in water phase. But for the stretch, because we know normally we wear textile or like in a dry environment, so we do that in vacuum to mimic that.
And also I don't know whether I've asked this before, but I have some like red lab thing. So maybe I think I asked before, like the TEV is kind of expensive, right? So what is the projected cost of your synthesis method for the fiber? And also how do you like because it will synthesize the peptide that will kind of make the culture kind of slimy. So how about the aeration problem in the culture, like scaling up, and also the removal of the host that can generate the fibers? So how would you plan to achieve that?
Yeah, the purification method is a very frequently asked problem. Actually, to be honest, I think we are not going to use the his-tag and the TEV protease in a later stage, especially when we are thinking about upscaling it into a industrial environment. As I've mentioned, actually we are actively searching on new purification system, and actually we have a quite feasible candidates now, not using tag proteins, not using his tag or similar tags to run columns to purify the proteins. And these ideas came from a 2018 iGEM team. It's using actually this project is quite hard to explain using few words, but it is like with a membrane, the E. coli can form a injectism to penetrate the membrane to express the protein at another compartment. So it is quite different from the his-tag purification system, and we are working on that.
Okay, so we also have another question from Simon. Simon, do you want to unmute yourself?
Yeah, sure. So yeah, I guess you can see in the chat room, yeah, very good presentation. You gave us a very good background on why basically you have very good motivation and why you're doing this. And I remember at the very beginning you mentioned the treatment for normal plastic is very expensive. That's why it causes pollution. So that makes me having a question at the very beginning on how does the cost compare between your production, let's say, is how would you imagine in a mass production the production of a fish swine to compare with the plastic that we're using? And yeah, a second question is I guess Kenneth asked it just now. That is a similar question for CityU. Like if you can go back to the lab, what would be the plan? I guess yeah, if you are not doing the joining the Giant Jamboree this year and this next year, then I imagine there will be a lot of experiment that you need to do. So yeah, I'm just curious.
Okay, I might answer the first question first. It is about the cost. Like as you might know, following my PPT, like we are using expensive lab techniques like TV protease, like his-tag. These are quite expensive. We don't have an exact number like we need how much extra cost to produce this IF, but as I've mentioned, we are actively searching a cheaper solution for purification, especially.
I guess you could do the comparison the other way around. I understand that it's very difficult for you to estimate the mass production of the project that you're proposing. How about you do it the other way around, like in a lab setting, for you to produce normal plastic, how do you compare the cost? Yeah, I understand you might not have the answer right now, but it will be interesting to include that number in your later presentation, either in the Giant Jamboree or whatsoever.
Yeah, we will look into it. Thank you very much.
Okay. So do we have any other comments? Oh, okay. Hannah raised hands. So Hannah, do you want to unmute yourself?
Yeah. Hi. First of all, same comment as Simon about the cost effectiveness. So I think it's also good if you interview maybe a manufacturer of clothing and then ask if what's the current cost of one single piece of cloth or the whole clothing or something like that. And then just curious about your future plan. And you mentioned that you will integrate a light sensitive production. Why is that light sensitive? Because light sensitive, wavelength sensitive promoters or constructs control are rare. So why are you choosing this particular method? And can it be done with other maybe chemical sensitive or other promoter circuits?
So basically the light sensitive thing is an interesting side project, which is like it haven't been researched much, but for now the idea is when you shine the same light, when you shine a light to the system will produce the same color as the light. So it will involve some kind of color mixing. For example, in the system there may be red and blue protein, and when you shine the purple light, which involve red light and the blue light, and it will produce the color mixing 50% to 50% of the red protein and blue protein, and eventually we hope to get the purple protein by color mixing. And this is just a proof of concept.
Okay, so it's just, yeah, I get it. It's an interesting side project, and it would be really fun to see the effect. But if you're upgrading it to upscaling it to an industrial level, wouldn't it be like more convenient to make it like place say 6:4 ratio of two chemicals, and then it will perform a color. So I mean, it doesn't seem to me that in an industrial scale it would be any different from shining light to it. So yeah, just a quick comment, and then about the future plan of wet lab experiments, maybe you will have to find a way to quantify the interaction between the alpha and gamma subunits and the interaction between it as not only two subunits and but the whole piece of fragment. So yeah, just find a way to quantify the set improvement of the fabric.
Okay, so do we have other questions or comments? If not, then we would really like to thank you all teams once again, and all the keynote lecturers once again for your presentations and sharings. So we'll now open the floor for the Q&A section. So if anyone has any questions, you can ask your question through the chat room. And please do mention the team or the person that your question is directed to. And you can also raise your hand, so we will unmute your audio and you can direct your question out loud to the respective presenters. Before that, does CityU team want to address Paul's question first? The one that we didn't have time to address. So Paul asks, have you checked that tetramer is the most abundant in nature? CityU team.
Okay, so actually plastic is a mixture of different polymer for different repeating units. So according to some research articles, if we use the pentamer or even longer polymer chain, then the polymer chain will be blocked by some amino acid, and the orientation of the substrate may change. So so we think that if we use a tetrameric form of the polymer, then this problem can be solved. So we decided to use the tetramer form instead of other longer polymer.
Yeah. Okay, thank you. So does anyone else have any questions? So just as a clarification, you can ask your questions to any of the teams which presented, both in the morning and also in the afternoon session. Because I think we have team representatives from all the 6 Hong Kong iGEM teams that presented their presentations today, both high school as well as undergraduates. So if anyone has any questions, or even if the teams have any questions to each other, maybe you can put it forth. Maybe we'll wait for 2 minutes to see if anyone has any questions.
Yeah. And I think Mr. Boeing is still here. So if you have any questions, can maybe also ask in the chat room. Yeah, we'll just wait for 2 more minutes. If not, then can end the meeting.
Okay. Okay, so if no one has any more questions, then thank you once again to all the keynote speakers, panel members, presenters, and participants for enriching our symposium with your knowledge, expertise, and participation. A special thank you to CityU for helping us design the brochure, and to HTUST for providing us with the technical support to stream the symposium through YouTube. We hope that all of you have gained some valuable insights from the symposium. See you in the giant jamboree. Thank you.
Yeah. Thank you.